chemical biology research, and especially, of course, highlighted by our two keynote lectures that will be given by Professor Benjamin Cravat. Now, the first talk of the seminar, and framing the whole program today, the last talk given by Professor Caroline Bertozzi tonight as the last talk of the symposium. I'll briefly introduce myself. So my name is Stefan Hacker. I'm currently an assistant professor at the Leiden Institute of Chemistry and was recently still a group leader at the Technical University of Munich. So that actually that move of mine also inspired us to look into this joint seminar series here, and to strengthen the connection between these two institutions that have a strong background and strong focus into chemical biology. So I don't want to take too much of your time today. I just want to give a brief introduction into the program, but also then some housekeeping remarks here, and then I will hand over quite quickly already to Ben Cravat for the start of the scientific program. So just briefly going through the program. So as you may have figured out, I now started with the opening remarks already, and directly after that, so by plan at 3, Ben Cravat will start with the first keynote lecture of today's program, telling us about activity-based proteomics and how we can use that to expand the druggable proteome. After that, Dorothea Fiedler will give her talk about their research into signaling by inositol pyrophosphates, and especially how we can use chemical tools in order to study these signaling processes. After that, we will have our first short coffee or bio break at probably around 4:15. So we have 3 of these short breaks, hopefully really ending up to be breaks in the program, otherwise at least allowing us to catch up with the time schedule. After that break, we will head into the second session with 2 talks. The first one given by Amy Weeks, who will tell us about how they can map protein modifications with spatial and temporal resolution. And then afterwards, Chu Wang will tell us about their efforts to profile the metalloproteome using one's chemical proteomics technologies, but also computational technologies. After the next short break, we will hit into the third session where Oina Yu will tell us what their group and also all of us can learn from electrophilic natural products in order to design covalent modifiers and probes to study biological systems, followed by the talk by Chris Parker, who will tell us about really new developments from his lab, which just came out as a preprint, where they use heterobifunctional molecules not for protein degradation, but to introduce protein acetylation at specific target proteins. After the last break, we will head into the fourth and final session of today's seminar, where the first talk will be given by Karen Beckes, who will tell us about her group's effort to expand the toolbox for chemoproteomic experiments, especially probably with a focus on cysteine profiling. And then we will end up the scientific program of today with a second keynote lecture given by Caroline Bertozzi, who will tell us about some exciting new developments by her group in targeted protein degradation, especially using their LITEC technology. To round up the program, Stefan Sieber is then going to give some closing remarks on today's seminar. So a couple of housekeeping remarks at this point. So as you are here, your link seems to have worked, and the good news is we will have this whole Zoom webinar entirely in this one meeting. So if you don't leave this meeting, you should be able to attend all sessions of today's seminar. You don't need any further links there. For the program, we've asked the keynote speakers for presentations of about 30 minutes, so that would give us time for about 15 minutes of questions and answers, and the other lectures will be, we asked about 20 minutes, so that there would also be 10 minutes of questions and answers. So you can see from that schedule that we quite generously planned for time for Q&As within the program. So we would also encourage every one of you to actively take part in that. So use the Q&A button, which should be on the bottom of your screen, to post your questions also already during the talks. And of course, for the hosts, the task will be to keep track of the Q&A function and then ask questions from there. To round up just these short introductory works, I want to thank a few people. So first of all, I of course want to thank the other organizers of these meetings, who are Monique Mulder, who's an assistant professor at the Leiden University Medical Center, Stefan Sieber, who's the chair for Organic Chemistry 2 at the Technical University of Munich, and Mario van der Stel, who's professor for molecular physiology here also at Leiden Institute of Chemistry. Also want to thank the administrative support by Jessica van Krimpen-Krijnoord and Barbara Seibold, and also want to thank Jan-Niklas Dünemann, who designed all these flyers and programs that you probably will have seen throughout the advertisement of the lectures. With that, I want to hand over already to the first talk, and I will stop sharing my screen so that Ben can probably already start sharing his. I'm going to give a brief introduction of Ben, although if you are here, probably he does not require too much of an introduction. So Ben Cravat is a professor at the Scripps Research Institute in La Jolla, California. I guess he's probably best known to most of you as a pioneer of the field of activity-based protein profiling in the beginning of that development, with a focus on studying serine hydrolases, so having the opportunity to really study not only the abundance of a protein, but studying how active actually those serine hydrolases are in first lysates, but then also living cells. And I think what stands out about this work is that while also many people, of course, directly apply the ABPP technology that they pioneered at that point, many of the technologies that factor into that have allowed many different developments also in related fields of chemical biology. Maybe for me, at least, recent very exciting developments in his lab is to look beyond certain specific families of enzymes and look very globally into all protein families, to all protein families, and discover which ligandable sites are still present in the proteome, and in this way make new proteins amenable to drug development. There, his group published large studies on profiling cysteines, but also lysines using covalent inhibitors and proteome-wide approaches, but also have profiled non-covalent inhibitors using photoaffinity-based labeling approaches to get a broad picture of the ligandability in proteomes. And this really has largely expanded our knowledge on what are ligandable pockets within the proteome. Besides his academic achievements, I think it's worth mentioning that Ben has also founded several companies, and especially I want to highlight Vividian Technologies that has recently been acquired by Bayer for their ABPP technologies. And I think this really nicely shows that also beyond academic interest, these technologies have really found broad application also in industry. So then we're really happy that you accepted our invitation to give a talk at this meeting, and I'm very much looking forward to your presentation. The stage is yours.
Thanks, Stefan, for that kind introduction. I assume you can hear me okay?
We can hear you. I can hear you.
Okay, great. Well, thanks for putting together this, to you and your colleagues for putting together this wonderful symposium. So as you mentioned, our lab's interested in developing chemical proteomic technologies to understand the functions of proteins in human biology and disease and develop ligands against those proteins. So I'm going to share two stories with you today, brief stories. Both actually could be summarized as using activity profiling to understand the mechanism of action of compounds that emerge from phenotypic screens. The first story will be squarely within the domain of druggable space. And through that story, I'll introduce some of the original concepts of activity profiling. And then we'll spend the second half of the presentation on extending activity profiling into maybe less classical small molecule protein interactions, and there also provide a bit of broader context about how we're trying to understand the broader ligandability of the proteome. So just one brief introductory slide that will be relevant to both presentations. We're going to be using activity profiling predominantly in this presentation as a way to read out the engagement of compounds that have been discovered in phenotypic screens. And so in this type of format, a compound or a compound library is added to a biological system. And the idea is that if you have a broad spectrum activity probe, it can sense where those compounds are, to what proteins those compounds are binding by a competitive displacement assay that can be applied directly in native systems. And you can read out the displacement of the activity probe by a hit compound from a screen in a variety of formats. This is just showing a gel-based readout where the signal is lost by, say, a fluorescent probe, but we'll mainly be using mass spectrometry approaches for that purpose today. And I think the reason why activity profiling has found a utility for this purpose is a fewfold. You'll see today that all the target and ligand discovery experiments I'll show are done directly in native cells and proteomes. So there's no need to have to recombinantly express proteins to understand their interactions with small molecules. And you also don't need a target-specific functional assay or even to derivatize or hit ligands from a phenotypic screen to discover their targets using this approach. And finally, for compounds that emerge from any type of screen, phenotypic or otherwise, activity probes are sampling, in some cases, 10,000, 20,000 sites in the proteome in parallel. So the ligands that they're evaluating get assigned not only as a potency factor against an individual target of interest, but a selectivity factor across the broader proteome, which can be helpful in understanding the specificity of compounds for current and then future pharmacological activities. Okay, so I think it's fair to say that activity profiling really started, as Stefan mentioned, in the domain of trying to develop probes that could target the active sites of large and diverse enzyme classes. And our lab and many others, as shown here on the slide, have been successful at achieving this objective. And our first story today will be how we're using activity probes combined with phenotypic screening to deorphanize members of the serine hydrolase family that play important roles in cancer cell biology. So serine hydrolases are sort of a prototype enzyme class, amenable and addressable by activity profiling. Very large, diverse enzyme family. About 1% of all the proteins in your body come from this class. Depending on the individual enzymes, they can perform pretty diverse chemistry, cleaving amide bonds in proteins, ester bonds in lipids, terminating the signaling function of neurotransmitters in your brain. So we thought if we could create broad spectrum activity probes for this enzyme class, you could explore a whole variety of physiologies and pathologies. Another neat feature of the family, which I think is also shared by many other large protein classes, is highlighted in this tree diagram where I'm ranging about half the enzyme class based on sequence relatedness and then color coding the enzymes as black or blue to designate whether they're characterized or uncharacterized, respectively. So there's a lot of blue in this graph, which means these are enzymes that we can assign based on sequence identity as being members of the hydrolase family, but we really have little understanding of their endogenous substrates, products, and functions. And as you'll see, that's going to be the focus of this first vignette. Now, a third feature of the hydrolase family, which is interesting, is membership does not require sequence or structural relatedness. I'm aware of at least 6 different folds, I think, that have evolved throughout nature to create an aberrantly nucleophilic serine residue in the active site of an enzyme to cleave amide, ester, and thioester bonds in biomolecules. So that's the one universally shared feature of the hydrolase family, that aberrant nucleophilic serine residue. And so if one wants to come up with a universal profiling strategy, you have to exploit that property of the enzymes. And when we started out with this effort, we had a good idea of how to do so. There were already well-established affinity labels, if you will, that electrophilic groups that could react with the serine nucleophile, shown here, a fluorophosphonate, to form this covalent phosphorus-oxygen bond. And so the concept we brought forward is you could create reporter tag versions of these fluorophosphonates. You could broadly profile the enzyme class, and these probes now can sample, probably interrogate close to 90% of the enzyme family. If the probe is biotinylated, you can enrich all those enzymes in parallel and identify them. Okay, so the story we're going to discuss today is using activity profiling to understand the role that specific hydrolases play in regulating the lipid modification state of NRAS. So I think to this audience, the RAS proteins don't need any introduction. They're major, in their mutated form, they're major oncogenic drivers of cancer in a variety of settings. Despite this knowledge, they've proven very challenging to address with drugs. There's several ways that this has been contemplated, and I think, again, as probably most of this audience knows, the greatest success so far, pioneered by Keyvan Shokat and Amgen scientists and others, is to trap the oncogenic G12C KRAS protein in its GDP-bound form and activating it with small molecules that covalently modify that oncogenic cysteine mutation. Here, we're going to be discussing an alternative strategy to regulate RAS proteins involved in reflecting changes or control over their localization. It ends up that the various forms of RAS proteins have a high degree of sequence identity across their entire protein stretch, except in the C-terminus, where they have a hyper-variable region that actually differentiates how RAS proteins are localized and associated with membranes. And that's shown here in this diagram, where you can take the classical oncogenic KRAS protein It has a polybasic sequence that helps facilitate this localization to the membrane. In contrast, NRAS, HRAS, and the KRAS4/8 proteins use a different approach. They have cysteine residues in their C-terminus that are dynamically modified by palmitic acid or lipid modified, and that helps them associate with the membrane and cycle through the cell for their signaling. And so that has led to the hypothesis that if one can disrupt the palmitylation cycle of oncogenic proteins like NRAS, you could potentially impair its signaling and perturb cancer growth, at least those cancers that depend on oncogenic NRAS for signaling. So our story begins with some pioneering work by Herbert Waldman's lab, where they discovered that these electrophilic beta-lactones, palmitostatin B and M, could block the depalmitylation of NRAS, stabilizing it in a palmitylated state and impairing its signaling. These beta-lactone type compounds are classic active site-directed covalent serine hydrolase inhibitors, and at that time it was inferred that their mechanism of action was largely through a couple of serine hydrolases called APT1 or APT2 or LYPLA1 and LYPLA2. These enzymes had been shown to be able to perform depalmitylation activities on proteins like NRAS in a biochemical assay. But in our experience and others, these beta-lactones are rather promiscuous electrophiles. So I think it was an open question at that point to what extent APT1, APT2 were the targets responsible for the lactone's effects on depalmitylation of NRAS. Concurrent with those efforts, Alexandra Atabekian, a postdoc in our lab at the time, now a professor at Scripps, Florida, had developed, using activity profiling, selective APT1 and APT2 inhibitors. And this allowed Elizabeth Conover's lab in Canada to test whether these inhibitors had an effect on NRAS palmitylation cycling. And unlike palmistatins, which nicely stabilized the palmitylated NRAS state, the APT1 and APT2 inhibitors in combination or separately did not alter NRAS palmitylation, nor did knocking down these two enzymes, providing the first evidence that perhaps other targets of the palmistatins may be responsible for regulating dynamic NRAS palmitylation. And that led the Conover lab again to use activity profiling, the fluorophosphonate probes I just described earlier, to begin to map additional targets of these beta-lactone compounds. And that led to a discovery of a subset of hydrolases that were all targeted in cells by the palmistatin beta-lactones. And a subset became of particular interest, three sequence-related enzymes called the ABHD17 hydrolases, which at that point were generally pretty uncharacterized. And indeed, Conover's lab showed that in a recombinant setting, each of the ABHD17 enzymes could depalmitylate NRAS. So the ABHD17 enzymes are interesting themselves. Brent Martin in our lab several years ago showed that these enzymes are themselves palmitylated at the N-terminus through this cysteine-rich region. They otherwise share very high sequence identity, almost 70% or greater across the three enzymes. And if you actually remove that N-terminal sequence that's palmitylated, you drive the relocalization of those enzymes to the cytoplasm. So that's the region of the proteins that are responsible for localizing the ABCC17 enzymes to the plasma membrane, positioning them in principle in the right place to control the dynamic palmitilation of NRAS. So we wanted to explore this further and try to understand which, if any, of these ABCC17 enzymes were responsible for regulating NRAS palmitilation in the context of oncogenic cancer signaling. And that actually was presented a rather daunting problem from a molecular biology perspective, because with 3 sequence-related enzymes that are expressed in pretty much every cell type, from a genetics perspective, it looked challenging to try to perturb and disrupt this family as a whole. So we took a different tack. We actually used activity profiling in collaboration with formerly a BI, but now Lundbeck scientists here in San Diego, led by Mike Nafakis, and also in collaboration with Kevin Shannon's lab at UCSF. Kevin's an NRAS or RAS signaling expert in general, to try to discover selective inhibitors of the ABHD17 enzymes. And so we were able to use activity profiling to discover these pyrzolureas as a good initial hits. These are also electrophilic compounds that act by a covalent mechanism. Here's your leaving group and what we refer to as your staying group that carbamylates the enzyme active sites. And through some medicinal chemistry, we were able to identify a solid, selective and potent chemical probe, ABD957, shows submicromolar activity against the ABHD17s, no cross-reactivity with the APT1, APT2 enzymes, and more broadly across human cell proteomes, rather selective, only a handful of off-targets that can largely be addressed by control compounds that I'll briefly mention later, and complete inactivation of all 3 ABHD17 isoforms in human cancer cells. So with these chemical tools, Radu Susio and Jarrett Remsburg, a student and postdoc in our lab, took on the challenge of trying to understand how does ABHD17 chemical inhibition affect dynamic NRAS palmitilation. And here we're going to use bioorthogonal chemistry pioneered by Carolyn Bertozzi. He'll be speaking later today in this context, using alkynylated fatty acids to follow the dynamic palmitilation state of proteins in cells. It's a pretty simple experiment. It's like basically a metabolic labeling experiment where you treat cells with the alkynylated fatty acid plus or minus your inhibitor, let proteins get palmitolated, and then chase with still in the presence of the inhibitor, but removing the alkynylated fatty acid. And so proteins that are dynamically palmitolated will lose their signal over time as they're being turned over, and the alkynylated fatty acid is replaced with palmitic acid, except those that are then stabilized by the small molecule inhibitor, which will then have preserved detectable signaling of alkynylation. And we're going to use 2 different cell models here created by Kevin Shannon's lab that are important to briefly explain. So these cell models are being generated in the OCI AML3 cancer cell line, where that cell line has an oncogenic NRAS driver. But we're going to knock down that oncogenic NRAS and replace it with a GFP-fused version of NRAS, so we can follow that protein as a GFP fusion. And also in another cell line, in the control cell line, replace that now with a GFP NRAS fusion, where the C-terminal hypovariable domain is now replaced with that of the KRAS. So it is no longer palmitolated, and that'll be a control cell line that should not be responsive to any of the effects of dynamic palmitilation on NRAS signaling. So we refer to those cell lines as ON and ONK, respectively. These experiments work quite well. Radu and Jarrett showed that you can see the dynamic palmitilation of NRAS with those alkynylated fatty acids. Here you see the signal prior to chase and the signal after chase. So within an hour, you're already seeing rapid depalmitilation of NRAS in the ON oncogenic NRAS signaling cells. In contrast, in the control cells that have this variant of NRAS with the non-palmitolatable C-terminus, there's no palmitilation. And you can see that the dynamic palmitilation of NRAS is stabilized by either palmistatins or a generic fluorophosphonate inhibitor, Hydrolases, shown here, basically almost completely. The ABCC17 inhibitor also stabilizes NRAS palmitilation, but partially, not fully. A control compound that targets the LYPLA1 and 2 or APT1 and 2 enzymes has no effect either alone or in combination with the ABCC17 inhibitor, again, providing it further support that those enzymes do not appear to be involved in regulating dynamic palmitilation of NRAS in this setting. So we were curious why the ABCC17 inhibitor was only capable of partially rescuing the palmitilation state of NRAS in this context. And we turned to mass spectrometry-based proteomics to get a deeper understanding of how these compounds were affecting the palmitolated proteome, if you will, of these cancer cells. This is basically the same pulse chase experiment I mentioned earlier, but now we're going to read out the palmitilation state of proteins by mass spectrometry rather than the gel that I showed you previously. So let me just walk you through what the data are here. So this is a graph presenting the effects of compounds on the dynamic palmitilation of proteins as read out by mass spectrometry. So on the x-axis, what you're looking at is the pulse chase experiment itself without any compound. So proteins on the right side of the dashed line here are dynamically palmitolated. So that they all undergo rapid turnover of palmitilation. And I should mention as a side note that a large number of palmitolated proteins are not dynamic. They're pretty stable. So it's important to run these pulse chase experiments to identify the subset of proteins that undergo rapid turnover in their palmitilation state. And then on the y-axis, as you move up the y-axis, you're looking at the subset of those proteins that are then stabilized in their palmitilation. A lot of proteins in this upper right quadrant marked in red, those are the proteins that are not only dynamically palmitolated, but in this case also stabilized in their palmitilation state by palmitostatin N. So we interpret from this result that most of the dynamically palmitolated proteins in the proteome are stabilized by this beta-lactone inhibitor. That's a very different profile that emerges from treating cells with the ADHD17 inhibitor, which now only stabilizes a small subset of dynamically palmitolated proteins, including NRAS and a couple of other proteins. The rest of the dynamically palmitolated proteins seem unaffected by blocking ABHD17 enzymes. And so we're curious, why might this be? What might these ABHD17 sensitive palmitolated proteins share in common? And as we began to look at these proteins in more detail, it became clear that what the ABHD17 sensitive proteins shared in common is a localization to the plasma membrane, and that the vast majority of other dynamically palmitolated proteins that were insensitive to the ABHD17 inhibitor were found in intracellular membrane compartments, as shown here. These were all stabilized in their palmitilation state by palmitostatin, but had no response to the ABHD17 inhibitor, ABD957. This began to give us some of our first hints that ABHD17 enzymes are not global regulators of the palmitilation state of proteins in the cell, but rather anatomically restricted, if you will, to the subcellular domain of the plasma membrane in their impact. So we wondered, and we also thought that this might be the mechanistic explanation for why ABHD17 inhibitors only partially stabilize NRAS palmitilation, because NRAS cycles, as I showed you earlier, through intracellular membranes as well, where it may be regulated by other palmitostatin sensitive enzymes. And so that then begged the question, what is the impact on NRAS signaling by just blocking its dynamic palmitilation at the plasma membrane? And we were able to see by measuring downstream phospho-ERK signaling that blocking ABHD17 enzymes did indeed impair, again, partially downstream NRAS signaling. So you can see that quite nicely here, that palmitostatin blocks NRAS signaling as read out by phospho-ERK disruption. So does the ABHD17 inhibitor. Neither compound has an effect in an NB4 cell line, which is a cell line driven by KRAS, right? So they shouldn't be relevant in this cell line where there's no dynamic palmitilation of the RAS oncoprotein. The MEK inhibitor, on the other hand, blocks both cell lines. So this is consistent with the partial blockade of depalmitylation correlating with the partial blockade of signaling in oncogenic NRAS cells. And that also correlates with a partial blockade of cell growth, as shown here, selectively in NRAS-dependent, but not KRAS mutant cancer cell lines. Finally, we found that Kevin Jannon's lab showed that the ABHD17 inhibitor it has an additive, if not possibly synergistic effect with the MEK inhibitor PD901, again, preferentially in cells that are driven by oncogenic NRAS. No such additive effect in cells that are driven by a variant of oncogenic NRAS that has the C-terminal hyperavailable region of KRAS and is no longer dynamically palmitolated. Okay, so wrapping up this first vignette, we believe the data that we've accumulated so far provides strong evidence that the ABHD17 enzymes are legitimate protein depalmitolylases, but show a plasma membrane restricted activity. And I should mention this is not the first example of an organelle restricted contribution of enzymes to regulating palmitylation. Some beautiful work from Brian Dickens' lab a while back showed that the ABHD10 enzyme regulates mitochondrial protein palmitylation. So sort of a similar story for a different organelle. We are still surprised by the restricted substrate profile for the ABHD17s, and we're trying to understand whether this just reflects localization of proteins to the plasma membrane that are palmitolated, or maybe there are even subdomains of the plasma membrane where these enzymes exert distinct substrate specificities. And finally, we'd like to understand whether ABHD17 inhibitors have potential as therapeutics in NRAS-driven cancers. This will require us to understand better how the RAS signaling is disrupted when you stabilize MS pulmonation. That's not actually totally obvious that would be the outcome, but that is what we see. We obviously, of course, like to further understand the partial effects that we observe, what other enzymes might be regulating dynamic pulmonation of NRAS, and what types of resistance mechanisms might emerge to ABH17 inhibitors over time. Okay, so let's move on now to extending the concepts of activity profiling to the broader mapping of the ligand-double proteome and a vignette about how this has helped us, again, in the context of cancer cell biology, identify a protein I think you'll agree is pretty much outside of classical druggable space and the role that it plays in controlling an alternative set of oncogenic driver proteins. So if you ask why our lab and Abide/Lundbeck and others have been so successful at creating covalent inhibitors of hydrolases that have progressed not only as chemical probes, but even drug candidates, the honest answer is we cheat. We cheat because the nucleophilic serine residue in the active sites of these enzymes is so aberrantly reactive compared to all other surface serines, maybe 7 orders of magnitude more nucleophilic. It gives the medicinal chemist a tremendous playground to come up with complementary electrophilic groups like the ones shown here and that I've already discussed that can target the catalytic serines and leave the rest of the serines in the proteome unperturbed. When one thinks about extending the concepts of covalent chemistry beyond the active site of a hydrolase, say to other amino acids like cysteine, you're presented with a different challenge and opportunity. So cysteine is certainly the most intrinsically nucleophilic residue in our proteins. And even the most nucleophilic variants of cysteine in our proteome are maybe only 1 or 2 orders of magnitude more reactive than all other cysteines that can be accessed by small molecules. And so this means if one wants to come up with selective chemical tools or drugs that target cysteines in proteins, you're going to have to probably balance reactivity and recognition elements of your compounds a bit more carefully. But it also presents an opportunity, right? Because it means that essentially any cysteine, if found in a pocket in a protein, could in principle be targeted by covalent chemistry. It doesn't need to have its intrinsic nucleophilicity augmented, if you will. And indeed, for those of you who are interested in covalent chemistry and drug development, you'll know that the vast majority of drugs that have been developed to date that target cysteines through covalent chemistry do so not by targeting catalytic cysteines in the active sites of enzymes, but rather by targeting, for instance, bystander cysteines in the active sites of enzymes like kinases. That's how drugs like afatinib and ibrutinib work. They target non-catalytic bystander cysteines in the active sites of kinases to form covalent adducts inhibiting those proteins. So this inspired us to think about whether we could more broadly look at the ligandability of covalent ligandability of cysteine residues throughout the proteome. And this is work that many former members of our lab have contributed to, including Ranthi Burapana, Chu Wang, who will be speaking later today, and Carrie Backus, who will also be speaking later today, who created a broad profiling strategy for evaluating how electrophilic small molecules can react with cysteines in the proteome. So in this platform, small molecules bearing cysteine-directed electrophilic groups and variable recognition elements are added to a cell or a proteome. And the idea is that these variable recognition elements will redirect these small molecules to different pockets in the proteome. And they'll bind those pockets. And if those pockets happen to possess cysteine residues, the electrophile can then latch on, forming a covalent adduct. And then one can read out those ligandability events using a broad spectrum probe like an itoacetamide, as shown here, which will react with thousands of cysteines in the proteome. Of course, except those cysteines that have been previously engaged by your electrophilic small molecules. They'll punch holes, if you will, in this broad proteomic profile that can be read out site-specifically by chemical proteomics. So this platform will not only tell you which proteins have reacted with these electrophilic small molecules, but also which cysteines on those proteins have reacted. And Carrie, in particular, pioneered the use of this technology to broadly profile the ligandability of cysteines in human cancer cells. And from those initial data sets, we began to get our first glimpses into the potential power of covalent chemistry to expand the druggability of the proteome. So Carrie identified well over 700 ligandable cysteines in her first analyses. And if you looked at the proteins that harbored those liganded cysteines, for the vast majority of them, this was their first evidence of interacting with small molecules by, say, having the presence or absence of those proteins in databases like a drug bank. When you look at the smaller sliver of the pie shown here, where Carrie was essentially rediscovering proteins that are known to be ligandable or druggable, you see a large representation of protein classes like enzymes, right? So proteins that we've had a pretty good understanding we can target with small molecules for quite some time. In contrast, the much larger slice of the pie, where this was the first evidence of small molecule interactions, you now see expanded representation of protein classes like transcription factors, DNA, RNA binding proteins, adapter proteins, again, providing some evidence that if one can integrate covalent chemistry with recognition, one can perhaps create chemical probes that can expand the scope of proteins that can be targeted by small molecules in our cells. So with that background, I'd like to share some work with you that's represented a very exciting collaboration between our lab and Janssen scientists, in particular K-On and Brahma Ghosh, where they were interested in trying to identify small molecules that could regulate the expression of the androgen receptor in prostate cancer cells. So as this audience probably knows, the androgen receptor is a major lineage driver of prostate cancer in humans. And as a result of this, there have been a variety of therapeutics that have been developed to target the androgen receptor, including this antagonist shown here, enzalutamide, coming out of initially Mike Jung and Charles Sawyer's labs at UCLA and Sloan Kettering, respectively. And like most cancers, when prostate cancers are put under the pressure of an antagonist of their major driver, the androgen receptor, they come up with resistance mechanisms that, in this case, are rather interesting and complex. So prostate cancers will sometimes overexpress the androgen receptor to limit the activity of antagonists, or create splice variants to the androgen receptor that no longer recognize a small molecule, or even make active site ligand binding pocket mutations that convert an antagonist like enzalutamide into an agonist to create a gain of function outcome. So these types of resistance mechanisms have motivated scientists at companies like Janssen to try to come up with alternative ways to suppress the androgen receptor in prostate cancer. So that background, we took our electrophilic compound library and shared it with Janssen so they could screen it in a very interesting high throughput screening assay, looking at a gene expression signature for androgen receptor, including the androgen receptor itself at the mRNA level. So we're asking these compounds to do something pretty significant, which is to lower androgen receptor gene expression within about 4 to 6 hours in cancer cells through this screen. Okay, so that's important because it'll be relevant to the mechanism of action we discover later. And we were able to identify compounds that in that brief period of time could suppress the androgen receptor mRNA in a concentration dependent manner with limited effects on cell viability, and you shouldn't have any effects on cell viability with a specific mechanism here within a handful of hours. And these ended up being these pyrazine chlorosetamides shown here. You follow over time, not only their ability to suppress the androgen receptor transcript, but also the androgen receptor protein. And if you follow those two events kinetically, you can see that the transcript for the full-length androgen receptor and its major splice variant B7 actually drop much more rapidly than the protein, giving a hint at the mechanism. The mechanism is probably not directly acting on the androgen receptor protein itself, but indirectly regulating this oncogenic transcription factor through transcript and transcript stability. As we began to explore the SAR of these hit compounds, I think we were quite fortunate in that we saw a rather exquisite structure-activity relationship. Very small changes to the compounds could either improve or completely eliminate the activity of these chlorosetamides. And even this one stereocenter proved to impart stereospecific effects in this assay. So the 10R variant is active at suppressing the androgen receptor. The 10S variant is completely inactive. And this was important, right, for target hunting, because this now gives us active and structurally related inactive control probes to read out cysteine engagement by these compounds in prostate cancer cells. And this is what work that was taken on by Stefan Kaufman, a postdoc in our lab. And Stefan used activity profiling, cysteine-directed activity profiling, to inventory well over 18,000 cysteines in prostate cancer cells with both active and inactive control compounds, looking for cysteines that were preferentially engaged by the active suppressors of androgen receptor, but not the inactive compounds. And that actually worked quite well. You can see here, and if you look at the sort of upper part of this graph, where we're identifying cysteines that are substantially engaged by these compounds, what we're looking for are compounds that are engaged by the targets that are engaged by the active compounds, but not the inactive controls. You can see that most of the cysteines mapped by Stefan that showed responsivity to the active compounds were also engaged by one or more inactive compound, except for the cysteine 145 in a protein NONO, which was engaged by all the active compounds, but not the inactives. So what is NONO? NONO is a RNA binding protein. It's a member of a small clan of 3 sequence-related proteins that form homo and heterotypic interactions. NONO is the only member of this 3-protein class that possesses a cysteine at position 145, indicating that the compounds would only interact with NONO and not its related proteins, SFPQ and PSPC1. So these proteins have been implicated in pretty much all steps of transcription and mRNA maturation, transport, stability. And so with the recognition that the compounds were binding an mRNA binding protein and that the compounds also suppressed antigen receptor transcript in cells, Stefan was eager to validate or to test whether NONO was indeed the target relevant to the mechanism of action of these compounds. And so he knocked NONO out of prostate cancer cells using CRISPR-Cas9 genome editing techniques. And this is where we began to get one of our first very interesting surprises. You can see here that Stefan nicely was able to knock NONO out of prostate cancer cells, but this had absolutely no effect whatsoever on antigen receptor protein or transcript expressions. You can see here by Western blot, the antigen receptor is at exactly the same levels in wild type and NONO knockout cells. What knocking out NONO did do, however, is completely block the ability of these compounds to suppress the antigen receptor, right? So you can see nice suppression of the antigen receptor by the 10R active ligand, no such effect in the NONO knockout cells. And this provided some of the first evidence that maybe these compounds are not acting as NONO antagonists, but rather maybe as gain of function or even neo-functional agonists acting through the NONO protein, requiring the NONO protein for their ability to suppress the antigen receptor. And as expected, the 10S inactive compound had no effect. And further supporting that mechanism of action, Stefan was able to reintroduce the wild type NONO protein into the NONO knockout cells and restore their sensitivity to the active 10R compound suppressing the antigen receptor and its splice variants. In contrast, if you reintroduce a cysteine 145 serine mutant of NONO that can no longer be bound by these compounds, you no longer restore their activity. So this, I think, provided very compelling evidence these compounds are acting through NONO to suppress the antigen receptor in a cysteine 145 dependent manner. So let me just finish up by sharing with you some of the directions we're now going to understand the mechanism of action better. So I think what Stefan, in collaboration with Janssen, has been able to show through integrated phenotypic screening and activity profiling is that this mRNA binding protein, NONO, is indeed a druggable protein through covalent ligands that target C145. And through doing so, these ligands can regulate antigen receptor expression in prostate cancer cells. As I've already alluded to, this seems to be a mechanism that involves a gain or neo-functional outcome on NONO because it requires NONO for the outcome. And supporting that hypothesis, Stefan has noted that when he treats cells with active ligands, they promote the clustering or speckling of NONO in the nucleus of prostate cancer cells, while the inactive control compounds have no such effect. So again, consistent with a gain of function type mechanism for these compounds. We're also interested in trying to understand more broadly how these neo-functional NONO ligands impact the broader transcript and protein profiles of cancer cells. And we've begun to do this looking both at proteomics and transcriptomics by RNA sequencing analysis. Let me just show you the latter, where you can see here in this graph, there are indeed a discrete set of transcripts that are suppressed by the active NONO ligands, but not the inactive enantiomer control ligands. And what's interesting is those transcripts are precisely the same transcripts that are regulated by the active ligands in a NONO-dependent manner. In other words, they are regulated by those compounds in wild-type cells, but not NONO knockout cells. And that can be seen here quite nicely with the antigen receptor transcript. The active compounds suppress that transcript. Inactive controls do not. NONO knockout on its own has no effect, but completely blocks the ability of the active compounds to suppress the antigen receptor. Again, all consistent with a sort of a neo-functional or gain-of-function mechanism. This also can perhaps best be displayed or visually by a principal component analysis, where if you look here, surprisingly, when you knock NONO out of prostate cancer cells, it not only has no effect on antigen receptor expression, it really has no effect, a very limited effect on the transcriptome more broadly. You can see here that the NONO knockout in wild type cells treated with DMO so look very similar in their global transcriptome profiles. In contrast, if you take the active compounds, you see that the active compound nicely discriminates between NONO, wild type, and knockout cells in this type of analysis. So there's a discrete set of effects on the transcriptional profiles of cancer cells with the active compound that is NONO dependent. And perhaps it's most interesting if you then look at the effects of the inactive control compound in NONO wild type and knockout cells. You see those cells now cluster based on their transcription profiles with the active compound in the NONO knockout setting. So we'd interpret this to mean that virtually all of the effects of these active compounds on transcriptional profiles of cancer cells occur through a no-no dependent manner. And when you get rid of no-no, whatever residual effects you're observing are observed with both the active and inactive enantiomers of these compound classes, and are no-no independent and maybe just general electrophile effects, so to speak. Okay, so finally, we'd like to develop more advanced compounds where we can study how targeting no-no might affect cancer cell growth, not only in culture, but also in vivo. And this is going to be challenging with these chloroacetamide compounds. You can see here in cell culture, you can indeed observe a stereoselective suppression of prostate cancer cell growth with the active compound in a no-no dependent manner. So if you get rid of no-no, those compounds no longer show this stereoselective blockade of cancer growth. But you'll also see that both the active and inactive enantiomers begin to kill cancer cells at pretty low concentrations, right? Low micromolar type concentrations. And this is just a reflection of the promiscuity of chloroacetamides at higher concentrations, and not something that we're going to be able to get around, I think, with that type of chemistry. So we've been trying to come up with ways to more broadly interrogate the ligandability of cysteines like C145 and no-no with compound types that are perhaps more progressible to advanced chemical probes and even drugs. And this has represented a very exciting collaboration with Stuart Schreiber's lab and Bruno Melillo here at Scripps, where what we're doing is really leveraging the diversity-oriented synthesis concepts of Stuart and integrating them into covalent chemistry and then chemical proteomics. So the library design principles here are important. Where the idea is we're going to create libraries of entropically constrained cores that are densely functionalized and stereochemically defined. This we believe would allow us to more rapidly discover useful chemical probes with tempered electrophiles like acrylamides. And Katja Vinogradova pioneered this work in our lab. She's now a professor at Rockefeller. And here's this one scaffold we've used, this triptyline scaffold, where Katja was able to show that one of these four diastereomers suppressed T-cell activation in a stereoselective manner, and she was then able to go on and rapidly elucidate the mechanism of action. That's a story for another day. I instead want to just finish in the last slide with showing you some of the principles that we've seen emerge, or the learnings that we've emerged from using these stereochemically defined diversity-oriented synthesis-inspired libraries. We like these libraries for a few reasons. One, if you do get stereoselective hits for cysteines in the proteome, they come immediately with built-in control compounds, right? As long as you demand that the ligandability events of interest are stereospecific, the enantiomers and diastereomers of your compounds can serve as built-in controls. We also believe that type of stereoselective reactivity is instant evidence of actionable pockets. If we can stereoselectively react with a cysteine in a proteome, we believe that's a site that can be progressed to a more advanced chemical probe. And finally, I should say that when we started applying this approach, I was concerned that by having these sorts of requirements of stereospecific interactions with sites in the proteome, the method might work, but it might land us squarely back in classically druggable space. In other words, if we have these sorts of criteria in place, would we only find stereoselective ligands for the active sites of enzymes, in which case it wouldn't have really helped us expand druggable space. Fortunately, that has not proven to be the case. We see no obvious bias towards classically druggable pockets in proteins when we apply these somewhat stringent filters of low micromolar engagement of sites in the proteome stereoselectively in cells. And just to finish, to show you some of those findings, here's just a smattering of cysteines that Katja and other members of our lab have found to show stereoselective reactivity with these triptyline acrylamides. And you can see these are adapter proteins, RNA, DNA binding proteins, and even in proteins like kinases, where we've been able to map stereoselective cysteines, we often find, like in the case of IKBKB, that this is a cysteine that's nowhere near the ATP binding pocket of the protein. It's actually in a protein-protein interaction site. So I think this is really encouraging from thinking about the broader opportunities of covalent chemistry to create advanced chemical probes and maybe even one-day drugs that target non-canonical sites and non-canonical proteins in the proteome. So I'll finish there and just think an outstanding group of students, postdocs, and collaborators with whom I've had the privilege of working. As I mentioned, Jerry Remsburg, Radu Sucio, and also Thomas Hannigan from our lab, working with UCSF colleagues Kevin Shannon, Naomi Zambetti, and others, and the Lundbeck team here in La Jolla, Mike Nafakis, to develop the ABH17 inhibitors and understand the role of ABH17 in NRAS pulmonylation and cancer growth. Stefan Kaufman, Jarrett as well, contributing substantially to our understanding of how these electrophilic compounds that suppress the androgen receptor operate through the RNA binding protein no-no in collaboration with Songjung Koo, Brahma Ghosh, Kayon, and others at Janssen. And finally, this wonderful collaboration with Stuart Schreiber and Bruno Melillo, led by Katja Vinogradova, and now Kristen Demeester, Evert Nijoman, and others, to try to understand stereoselective electrophile reactivity in the proteome as a conduit to advance chemical probe development. And so I'll finish there and be happy to try to answer any questions you might have. And thanks for your attention.
Thank you very much, Ben, for this really insightful talk, to really interesting stories, I think, that you presented. And there are already some questions coming in, so please, general attendees, put your questions in the Q&A function in order for them to be asked. So the first question was from Stephen Helst. He asked about the first part of the talk. Why do you think that many proteins are stably punctuated and only a small subset is dynamically regulated?
Yeah, Steve, that's a great question, Steve. We don't honestly know the answer, but it is surprising, right? I mean, if you look at our lab's data and other labs' data, Howard Heng and others, a very small fraction of palmitoylated proteins are what we would call authentically dynamically palmitoylated. So there's a couple answers to this. One would be biologically interesting and one would be less biologically interesting. So the more biologically interesting one would be that there really are proteins where enzymes are putting palmitate on, but they're the sites that they're palmitoylating are somehow evolved to be more stable, right? So those proteins do not undergo dynamic turnover, and that's the purpose of the role that those palmitates play. I think the less exciting answer might be that there's a lot of fatty acid coenzyme A floating around in the cell, and there's a lot of cysteines, and some of those events may just bump into each other over time and create low stoichiometry palmitylation events that the cell just kind of ignores, right? That they're just there as like bystander modifications of proteins that don't play a major role in how those proteins function, but the cell just tolerates them because they're also not a costly outcome. And my sense is some of it is actually the latter, but of course that's just my speculation. So we don't honestly know the answer to the question, but it is true that I would say probably at best 10% of the palmitoylated events that we've mapped in the proteome are authentically dynamic.
So question by Stefan Sieber. Great talk, Ben. Impressive hit by your electrophilic library. It looks like that the hit for NONO is already quite potent. Do you know how it binds to NONO, for example, putative binding pocket, and do you have plans for rational optimization?
Yeah, that's a good question. I should have mentioned this, but the cysteine that NONO binds is actually in this beautiful pocket that is at the protein-protein interface and also predicted RNA binding interface of the proteins. So as these RRM and RM2 domains are, they're the RNA binding components of NONO. So, Stefan, when I look at this site, it again begs the question of whether we're sort of rediscovering what nature is already using as a mechanism to regulate NONO. It looks like a pocket where an endogenous, maybe electrophilic metabolite could bind, and that's something we're trying to explore now. I do believe this pocket, anytime we see that type of stereospecific interactions, I do believe the pocket is druggable. The question really is, do we approach this, as you suggested, through trying to rationally optimize the ligands we have in hand, or maybe look more broadly at chemistry that can engage that pocket? I really do want to get away from chloroacetamides if we can as ligands for this pocket. I'd prefer to find an alternative electrophile that maybe had a bit more stability, but we're exploring all options right now. And but you've seen the chemistry of where we're at right now. We've explored very little so far. And I think maybe one of the more interesting questions that we'd like to ask downstream is, do all ligands that bind this pocket produce the same type of effects on the transcriptome? Or might it be that different ligands that bind this pocket produce different subsets of outcomes on transcriptional regulation? That would be quite exciting, right? It would almost become like IMiDs binding E3 ligases, where you could tune effects on different transcripts by different types of chemistries binding the same pockets. Those are the types of ideas we're trying to pursue going forward.
Great. Yeah, that would be really exciting. So
that's just theoretical. We don't have any data to support that right now.
Yeah. I have one anonymous question. So great talk, thank you. How do you typically select the compounds for phenotypic screening? I'm interested in the idea of exploring a wider area of chemical space.
Yes, that's a great question, and that's an evolving answer. So I would say that we now are extremely committed to building next generation libraries that embed stereochemistry in almost every compound that we make, because it's just proven so useful for rapidly deconvoluting targets. We got very lucky in this NONO example because our library that we screened at that moment was not very, very rich in stereochemically defined small molecules. And it just happened to be that the hit compound had a stereocenter that ended up being capable of discriminating the mechanism of action. And so, the nice thing about covalent chemistry, if you embed stereochemistry and have good structure-activity relationships in your libraries, you can push those compounds pretty hard in phenotypic screens, right? They don't need to be target specific. They just need to have a structure activity relationship that is robust enough that you can distinguish the targets that matter for your phenotype. And one other comment on that, I would say that I made passing reference to is the only reason this screen worked at all for discovering the no-no ligands was because we were able to screen so quickly, right? We looked at transcriptional changes in 4 or 6 hours. If we had waited 24 or 48 hours, I think the cytotoxicity effects at the concentrations that we screened at for those compounds would have been prohibitive to identify that type of stereoselective mechanism. So, as you think about phenotypic screens, especially with electrophilic compounds, I always encourage people to consider screens that can be done rapidly, like within 8 hours or less in cells. I think that really allows you to push the concentrations of those compounds without ending up in the domain of causing cytotoxicity.
Maybe one last question. Really interesting talk. Thank you. Do you think resistance mechanisms would be less likely with the no-no ligands since you're not antagonizing androgen receptor directly?
That's a good question. I'd like to believe that, but I probably don't think that's the case. I mean, I suspect that the most obvious resistance mechanism you could get here would just be to mutate the cysteine into serine and no-no, and the ligands would fall off and become inactive. And I don't see why that wouldn't occur in this context as well. I mean, in some ways, you could argue that this mechanism is a little bit similar to how, maybe some of the spliceosome inhibitors operate or modulators, and those mechanisms of inhibition have been amenable to resistance mechanisms in cancer cells where the mutations disrupt ligand binding. So I don't think we're going to be able to avoid any resistance mechanism here, even though we're not directly targeting the androgen receptor. Now, it could be different resistance mechanisms which may allow for combination therapies in a way that down the line proves to be more durable for therapy. But cancers are pretty capable of resisting almost any targeted mechanism, in my view.
Great. Thank you so much again, Ben. Really insightful talk. Very nice discussion. So let's move on to the next talk of the program. Maybe Dorothea can already start sharing her screen. So I want to start by also give a quick introduction to Dorothea. So Dorothea studied chemistry at the University of Würzburg and did her diploma thesis already in Berkeley, where she then did also her PhD, still very much in inorganic chemistry, working on host-guest systems. And for a postdoc, moved on to the lab of Kevin Shokot at UCSF, where actually she started working on biological signaling systems. After that, she went on as an assistant professor at Princeton University, and 2015 got recruited back, I guess, to Germany to become a director of the Leibniz Forschungsinstitut für molekulare Pharmakologie in Berlin, and also a professor at the Humboldt University in Berlin. Her independent work is pretty much focused on the biological role of inositol phosphates and inositol pyrophosphate, and I think over the last year she has contributed to synthesis of quite a lot of great tools in order to study phosphate biology, and especially lately she's brought out interesting tools that allow us to identify and characterize binding proteins of these highly phosphorylated molecules. Dorothea, we are very much looking forward to your talk. The stage is yours.
Okay, thank you. I'm assuming you can see my proper slides.
Yes,
after we had some technical problems earlier. Very good.
Yeah.
Okay, so let me start by thanking Monique, Mario, and the two Stefans for putting together this wonderful symposium. And as Stefan already told you just a second ago, my group is interested in signal transduction, and in particular, our major fascination lies with these highly phosphorylated inositol poly and pyrophosphates. So the inositol poly and pyrophosphates, they are related to the phosphatidylinositols. And oftentimes the inositol phosphates are actually derived from the phosphatidylinositols. So PIP2 here, which is anchored to the membrane, can be cleaved by phospholipase C, and that generates IP3. This is a well-characterized second messenger, important in calcium signaling. But IP3 can be further phosphorylated by these small molecule kinases, IPMK and IPPK, to ultimately provide an inositol phosphate that carries 6 phosphoryl groups. So all 6 hydroxyl groups are now phosphorylated. Quite recently, an alternative biosynthetic route was described by Adolfo Sayardi, where they showed that glucose can be converted into inositol monophosphate, which can subsequently be further phosphorylated by ITPK. But again, also here we ultimately end up with this molecule Ip6. And so this is where I think our general fascination becomes even greater, because as if 6 phosphoryl groups are not enough, there are small molecule kinases called the Ip6 kinases and the PPIP5 kinases that add additional phosphoryl groups. In the case of Ip6 kinases, they do this at the 5 position, so generating this small molecule 5-PPIP5. PPIP5 kinase adds it at the 1 position, and so the combined action of Ip6 kinase and PPIP5 kinase ultimately leads to the formation of Ip8. So you have a 6-membered carbon ring that now carries 8 phosphoryl groups. And I think that the chemists in the audience immediately appreciate how much negative charge this really is, how many phosphoryl groups. And then also from a biosynthetic perspective, each phosphorylation requires ATP. So this is some dedication of the cell to really be generating these metabolites. The inositol pyrophosphates have been detected basically in every eukaryotic system tested so far, and the enzymes involved in their biosynthesis are very well conserved. And especially the inositol pyrophosphates are known to respond to specific stimuli and turn over rapidly in cells, implicating them in signaling processes. And this general scheme here is a bit of a simplification. In reality, there are even more different inositol poly and pyrophosphate species that can be found in different eukaryotic systems. So while certainly fascinating from a structural perspective, people have investigated inositol pyrophosphates using genetic methods. And there's, I would say, a landmark paper from Sol Snyder's group where they knocked out Ip6K1. And they characterize these knockout animals in great detail. And one really striking phenotype is that the knockout mice, both female and male, are resistant to high-fat diet-induced obesity. So you can put them on a high-fat diet, but they do not gain weight. Takeda Pharmaceuticals looked at mice lacking Ip6K3, and while those mice did gain weight, they lived longer. So yielding a different picture. And then, in my opinion, it gets even more complex because it was also shown that when you delete Ip6K2, so again, we're within this kinase family here, this kinase now seems to be relevant in tumor cell migration. So what was shown, again, by the Snyder group is that cells lacking Ip6K2 have a much lower propensity to migrate and grow. And also in xenograft tumors grown from Ip6K knockout cells, these mice showed a much lower propensity to form metastases at distant sites. So the point I'm trying to make is that this is a complex interplay of different metabolites, and genetics has taught us a great deal about what these molecules may be playing a role in. This is really just an abbreviated list here of the different phenotypes that have been associated with these highly phosphorylated metabolites. But what, of course, is missing is, to some extent, a connection of these molecules to these specific phenotypes, right? How do these molecules work at a molecular level? And this is a difficult question because evidently there seems to be tissue-specific production of certain metabolites or organelle-specific production. And then, of course, there's the question of ligand specificity, right? How is it possible that certain proteins will recognize 5-PPIP5, but maybe not the precursor Ip6 or the highly structurally related 1-PPIP5? And this question is further complicated because so far there have been different modes of action proposed for these inositol pyrophosphate messengers. So I think the intuitive one here is allosteric regulation, where you can imagine these molecules bind to specific target proteins and thereby somehow alter their activity. But the example from the Snyder group, for example, with regards to weight gain and insulin resistance here, has to do with competition with PIPs. So in some cases, the inositol poly and pyrophosphates can compete with the phosphatidylinosytols for lipid binding domains. And in this case, this is how insulin signaling was affected in these knockout mice. Other reports have nicely demonstrated how these highly negatively charged molecules can basically act as a molecular glue to bring together two positively charged surfaces. And also this, of course, is an important mechanism of action. And then lastly, these high energy metabolites have been proposed to participate in unusual phosphoryl transfer chemistry that ultimately leads to the installation of these pyrophosphate groups on proteins. So basically, I think everybody in the field agrees that it is quite difficult to decipher these signaling networks. And really the bottleneck to all of this is finding inositol pyrophosphate interacting proteins and understanding the specificities in these protein ligand interactions. And this is something that we felt we could do, and that has accompanied also my lab from the very beginning. So actually the first graduate student in the lab, Ming-Xuan Wu, he decided that he would take a classical chemical biology approach and use affinity enrichment to see which proteins physically interact with inositol pyrophosphate. So he synthesized this molecule here. It's 5-ppIP5, but the pyrophosphate group here has been replaced by a bisphosphonate group. So we have a methylene group here in place of the oxygen, and this renders this molecule resistant to enzymatic hydrolysis. And then here at the 2 position, he installed a soluble PEG linker and then tethered this onto agarose beads. It's sort of in an abbreviated form. You'll see this later in the talk as this little scheme here. And then he exposed this to cell lysates from Saccharomyces cerevisiae. He also made the corresponding reagent with Ip6 tethered. And we looked globally and found some interesting binding partners. One thing we were really after was finding a dedicated inositol polyphosphate binding domain, because much in contrast to the phosphatidylinositols, that had not been described to the soluble and highly phosphorylated inositol phosphates. And we did find several proteins in yeast that contain this SPX domain. This is a domain that proteins involved in phosphate homeostasis contain. And gratifyingly, sort of simultaneously as our paper was being published, there was beautiful structural work from Michael Hutton's group that showed that indeed the SPX domain binds to Ip6 in this case, and there is a number of lysine residues here that form a lot of electrostatic interactions with the negatively charged Ip6 molecule. So again, part of the mission was accomplished. We found this interesting domain, but then on the other hand, when we compared our data that we had obtained for 5-ppIP5 and then compared that to Ip6 proteins, interacting proteins, it was quite disappointing because there seemed to be very little specificity with regards to which proteins would physically interact with one or the other molecules here. And we made the linker attachment responsible for this, because if you look closely, you'll see that this 2 position here is the only axial position in the molecule. And this is where we put the linker. It was for synthetic ease, but it also perturbs binding here of this axial phosphate group to target proteins. And so we think we may have perturbed some of these interactions quite significantly. And then the idea was, well, maybe we should be attaching the linker at different positions or try multiple positions at the same time. But we put that on hold because the chemical synthesis was a bit tedious. Fortunately, we had a wonderful postdoc in the group, Sarah Hostachi, and she came up with these combined phosphate ester phosphoramidite reagents. So they contain a phosphorus 3 and a phosphorus 5. Within the same bisphosphonate here. And these reagents were first developed by the Filipov group for generating ADP ribose analogs. And Zara basically made these P5, P3 reagents or PCP amidites compatible with our inositol phosphate chemistry by switching out the protecting groups here and characterizing these reagents well. And just as a quick illustration that this approach worked well for her, what you can see here is that even at this highly sterically encumbered 2-hydroxyl group, she could append the PCP amidite in good to really excellent yields, depending on the protecting groups here, and generating these PCP analogs in really high yields that we also use for a lot of different applications in the lab. And so this then in turn motivated us again to revisit the alternative linker attachment. And this was taken on by David Foucault, who's currently a graduate student in the lab. And I'm not going to walk you through the whole synthesis here, but the important part is that we can start from this readily accessible intermediate here, and again append the PCB-amidite at this 2-hydroxyl group, and then we can remove the acetal protecting group and subsequently just attach one equivalent of linker either at the 1 or the 3 position. We then carry through the whole steps, and then at the end we have a 5-PCP inositol pyrophosphate here that carries a linker either at the 1 or the 3 position. And this linker in turn can again be immobilized onto a resin, and then we'll have an affinity reagent with the linker at the 1 or 3 position attached. David also generated the corresponding Ip6 reagent, and with that he then went on and validated his new affinity reagents and compared them also to our previous affinity reagents using known binding proteins. And so NUT3 here, for example, is a phosphohydrolase that would typically cleave off this beta phosphoryl group, and it has a high affinity for both Ip6 and 5-PDIP5. And then here, XPR1 is the one human protein that contains an SPX domain. And you can see when we apply these proteins to our reagents, they're removed from the solution. The control reagent, which has the same resin and the same linker, but just a phosphoryl group up here, does not bind to these proteins. And the same is true for our new reagents. And subsequently, we elute with an excess of Ip6 or 5-PPIP5, and the proteins come off again. So this all seems to be working according to plan. So then with these reagents, David wanted to set up a proteomic experiment, and he decided to ultimately generate an affinity reagent in which we had the 5-PCPIP5 immobilized at the 3 different positions, 1, 2, and 3 in equal amounts. So we mixed the old 2 linker reagent with the 1,3 reagent in the right stoichiometry. He did this for Ip6 as well. And then we applied cell lysates from HEC293 T cells. Just out of curiosity, we looked at our old reagents again as well. We washed, we eluted, we digested, and then used label-free quantification to assign which proteins were retained by these resins by mass spectrometry. And here is a volcano plot for the 5-PCPIP5 reagent versus control. And there are many proteins that were specifically retained by this reagent. And in a first pass of analysis, what made us happy was to see that many of these proteins made sense. So again, there are these phosphohydrolases that would use this as a substrate. There's also synaptotagmin, a known binding protein both for Ip6 and 5-PPIP5. Also, what is important to say is that qualitatively, at least, these multiplexed reagents performed better than the old reagents. We got better coverage. We got somewhere close to 400 proteins that we could identify. And also when we used HCT116 cell lysates, we got very similar results. So all this looked like a stable analysis. And of course, we can ask some general questions. For example, about the domains that preferentially interact with these highly phosphorylated inositols. And what we see here in the mammalian cell lysates is that this competition with phosphatidylinositols seems to be quite a relevant mechanism. So the top 2 domains are the C2 domain and the plexrin homology domain that is contained in proteins enriched with these reagents. And that indicates to us that there is indeed a fair amount of competition going on with phospholipid binding. But I think the major question that we wanted to ask is there now specificity in these proteomic data sets, right? Are there proteins that specifically interact with Ip6 or 5-PPIP5? And so in a first comparative analysis, we asked which proteins preferentially interact with Ip6 versus 5-PPIP5. And we're really excited to see that there is a good number of proteins that exhibit specificity. Here we were really pleased to see that we found a number of proteins that made perfect sense to us. So these are proteins that are part of the COP9 signalosome, or CSN. And this is a denatylase holoenzyme that regulates the colon ring ubiquitin ligases. And Feng Rao's lab had shown before that the interaction of the CSN with colon ring ligases here required Ip6. So there's a crystal structure where Ip6 sits at that interface and is important for bringing these two components together. And interestingly, what they also showed is that if Ip6 is further converted to 5-ppIp5 by the inositol kinases, this complex formation is destabilized. And we were really pleased to see that this was captured well by our proteomic data. Nevertheless, I think the analysis the other way around is really what we were after. And so I'm happy to share this here, that when we ask which proteins preferentially interact with 5-ppIp5 versus Ip6, we also got a number of really interesting hits. And also it was nice to see that they seem to be falling into certain groups. So everything shown here in blue are inositol 5-phosphatases. And David here also validated this by Western blot. So what you can see here for INPP5K, for example, that what comes off the beads here at the end is that it is retained really well by the PCPIP5 beads, not so much by the IP6 beads. And then when we add competing ligand, this interaction is disrupted. And the same is true for some of these other 5-phosphatases. These are lipid phosphatases. So most of them act on phosphatidylinositols and remove the phosphoryl group at the 5 position. And they target distinct intracellular pools of phosphatidylinositols with differing global impacts on phosphatidylinositol levels. And they all contain this 5-phosphatase domain. So the question was, is this really the domain that is physically interacting with the inositol pyrophosphates? So this is the general structure here of INPP5B. This is the example that we picked out. It does have a pH domain, but some of these other 5-phosphatases did not. So we focused on this 5-phosphatase domain. David expressed and purified it, and again, could show really nicely that indeed the 5-PCPIP5 reagent retained INPP5B phosphatase domain and could be eluted off from that resin again. So then, of course, comes the mechanistic question, right? What does this interaction do? And the first thing we tested was whether 5-ppIp5 could be a substrate for INPP5B, which it is not. But it turns out that 5-ppIp5 can inhibit the dephosphorylation of the lipid, and it does it quite potently with an IC50 of around 1.5 micromolar. And then by comparison, IP6 is a much weaker inhibitor. So this was all well and good and exciting, and certainly it gives us a really fascinating biological direction to go into. But what this also illustrates, again, I think with these pull-on pictures and also maybe these IC50 curves, is that we have no good measure of how strong these interactions really are. And especially if you want to do comparative analysis of IP6 versus IP5, IP7, et cetera, we should be thinking about a biophysical characterization. And people have used classical methods like NMR or ITC, but they can be tricky because on the one hand, they require relatively high protein concentrations. And then also, when you bring together this highly negatively charged inositol polyphosphate with a cationic binding site, there's a lot of charge neutralization, and our number one problem tends to be precipitation. So this motivated us to think about a different type of assay to measure inositol phosphate binding interactions, and we arrived at surface-based methods, and specifically surface plasmon resonance spectroscopy. And so the idea is the following: we have an inositol phosphate, in this case IP6. We have it already derivatized with the linker at the 1, 2, or the 3 position. Shown here is just for the 2 position. And we could then, at that primary amine at the end, install a biotin. We use a gold surface that is coated, and as we flush analyte, in this case protein of interest, over it, we can measure association rates and dissociation rates, and from that also determine the equilibrium binding constants. We did this in collaboration with Michael Hutton's lab in Geneva, and his postdoc Daniel Kuto worked together with my student Annika Richter. And they first used a known interactor to make sure that the system really works as we expected. And so in this case, again, we use this SPX domain, and I hope you can see that this gives nice association and dissociation curves, and from that we can measure an equilibrium binding constant. In the case of the 1,3 linker IP6, this is a little bit lower at around 470 nanomolar, and this particular interaction was characterized with ITC, and this value is quite close to that that have been determined in the past. So that made us confident that this was a good method in principle, and Annika then went on to prepare the corresponding 5-PCP IP5 reagent with the linker at the 1 and the 3 position, just because we do now have new appreciation for this 2 position here, and used purified IMPP5B, the phosphatase domain, to look at the interaction. And what she found is that, yes, indeed, we do see nice strong interaction with IMPP5B. The equilibrium binding constant is around 4 or 5 micromolar. And this is actually right on the range of concentrations that these molecules occur naturally. So IP6 is more abundant, maybe at 30 to 50 micromolar in cells, but 5-PPIP5 is the most abundant pyrophosphate, and this is in the single-digit micromolar range in mammalian cells. Her first tries, Annika's first tries to measure an interaction with IP6 here failed, and this is because the affinity is so low. So we need to go in with higher protein concentrations, but we really can say with certainty now that IP6 is not a strong interactor with this phosphatase domain. And so again, this gets us back to these mechanisms. We still can't say with certainty whether the inositol pyrophosphate competes for the substrate binding site of the lipid. But I don't know, I still don't understand why IP6 wouldn't do this competition. So my feeling is this is an allosteric regulation. And we're fortunate that my colleague Hauke here at the FMP is a phosphatidylinositol expert. And so we're teaming up with him to now follow up in a physiological context and understand what this interaction means. And so if I have one last minute, I want to give you one last example that we're also excited about following up. And these are these ribose phosphate pyrokinase interacting proteins. So PRPS1 and PRPS2 are ribose phosphate pyrokinases. So they convert ribose 5-phosphate to phosphoribosylate pyrophosphate. And that is essential for de novo purine biosynthesis, which in turn is required for supplying nucleotide precursors. And also this interacts nicely with our resin. We see a strong interaction using SPR with the binding constant of 4 micromolar, which we think could be physiologically relevant. And what, again, is particularly exciting about this protein is that we have cells that are lacking IP6 kinase 1 and 2, and we know they display a significant accumulation of intermediates in purine biosynthesis. So there is a phenotype in purine biosynthesis. And this is the reason why this is another target we would like to follow up on. And so what I would like to leave you with is that the inositol pyrophosphates can access distinct modes of action, and they can target a variety of different protein domains, as we've learned in mammalian systems. The inositol pyrophosphates seem to be metabolic regulators or influence different metabolic processes. We found this intersection with phosphatidylinositol metabolism and also de novo purine biosynthesis. But certainly this better mechanistic picture that we've gained from our affinity reagents and now also the biophysical measurements really needs to be complemented by functional analyses. And this is what we're trying to accomplish in the future. And so with that, I would like to thank my group. I think I've mentioned everybody along the way. Ming Xuan and Zara, former co-workers. Henriette was a master's student who started working on these biotinylated inositol phosphates. And then David here and Annika are still in the lab. And yes, I'd like to thank our collaborators, especially Michael Hotwan at the University of Geneva, and all of you for your attention, and I'd be happy to take any questions.
Thank you very much, Dorothea, for this very nice talk. Actually, there are a couple of questions already, so I'm going to start with one question by Ben Cravat. Great talk, Dorothea. Considering the polarity of these inositol polyphosphates and their corresponding cationic binding pockets, How do you foresee the discovery of chemical probes that perturb these interactions? Will these polar pockets prove difficult to target with conventional small molecules? And if so, might allosteric mechanisms be a more productive path?
Oh, okay, good. Well, I mean, I think this is a great question. In some ways, yes, it would be great if we could target these polar pockets. I agree. It seems really unlikely that we find a good inhibitor in a conventional library. One thing that I actually wanted to talk to you or a related person to is that I think a lot of these pockets, they're full of lysine and arginine residues. And so I think there are some severely pKa perturbed lysine residues in there. And so I think that may be an angle to get at that, because I almost can't think of more highly cationic pockets. And I think if we could take that to our advantage, that may be a way of getting at this question in the long run.
Thanks. So I will take one more question from the chat or from the Q&A. Thank you for the great talk. The competition with pH C2 domains is very striking. Could some of the physiological effects be explained through regulation of PIP kinases on the plasma membrane, or do PIP kinases have cytosolic functions that are activated in this fashion?
Yes, so we actually, in our proteomic data, we do see a lot of PIP kinases that will physically associate with Ip6 and also 5-PPIP5. And I do think there is a fair chance that there is this competition going on, and that it is an important factor regulating the localization of these PIP kinases. To what extent? Yes, this is specific to the C2 domains. I can't say with certainty. I think for that we would need more data, but it would be great to just start looking at localization data of these PIP kinases, for example, in response to pharmacological perturbation of inositol kinases or inositol pyrophosphate biosynthesis in order to also gain more insight there. I do think it is an important regulatory mechanism overall.
Thank you so much. So there's one very technical question still in the Q&As. Maybe you can answer that in writing during the break or the further continuation of the program. Thank you very much to both Ben and Dorothea again for very nice talks. We will now go into a first short 15-minute break. So we will reconvene at 4:30 Central European time with the second session with talks by Oina Yu and by Amy Weeks and Chu Wang in the second session. So thanks again to the speakers and everyone asking questions, and see you again in about 15 minutes.
Thank you.
Dorothea, if you can stop screen sharing, then I can put the general slide on.
Yes, of course. That worked, right? Good.
can currently see your screen, but in the general PowerPoint mode.
Can you see it now, Stephen?
Yes, it's in the full screen mode now. Works. And I can also hear you all right, I think.
Okay, thank you. Yeah,
Perfect.
good to see you.
Sounds good.
Okay, I'll start sharing.
Perfect, thanks.
What's going on, Shu?
Hi, Ben. How are you?
Good. How's life?
Yes. Yeah, it's great. Sorry, I came in a little bit late because my other computer got frozen. So while I opened it, and then you already started your talk.
Great to see you. Oh yeah, that's
great to see you. Yeah.
Everything good.
Yeah, I think it's everything going all right. It's just you know, the COVID-19 thing here has very strict policy, so it kind of prohibit a lot of like international travels. So,
right.
But other than that, it's good.
Are you guys generally vaccinated already out there, or how's that going?
Yeah. So I think most of the population in the cities, and especially like Beijing, is a capital, right? So I think a large percent of the people already vaccinated and already twice. So they're debating whether they should also do the third vaccination for this Delta variant.
Yeah, I mean, I don't see why not. You know, it's I guess if there's a way to distribute the vaccine to other areas of the world that don't have it, I would ban it. But if it's vaccine, it's just going to sit around and not be distributed. Seems like Israel, the data is pretty clear that it definitely helped. You know.
Yeah, because they are also trying to get ready for the Winter Olympics, right? So which will be held like February next year. So it's coming pretty soon.
That's right. Because the Summer Olympics are a year late, so the Winter Olympics are next year. I forgot about that. That's like 4 or 5 months, right? It's not even that far.
Yeah, exactly. So that's also like the first time Beijing to host the Winter Olympics. Actually, it's the first city to host both Summer and the Winter Olympics.
Oh, I didn't realize it was Beijing. It's actually Beijing that's hosting it.
Yeah, it's Beijing.
Are there mountains close to Beijing?
There was actually on the northwestern part of China, Beijing. So on the northwest, so they built a high-speed railway. So for the skiing.
Okay, so not that close, but you can get there. Got it. Okay.
Exactly. So actually, the high-speed railway only took like 30 minutes. So so it's very convenient. And also they have all the like ice skating, all the others within the cities. So.
That's great. I'm glad you're doing well. So it's been a long time.
Yeah, I haven't seen you for a while. Actually, remember like that's last time I was in San Diego. That was right before the outbreak of this COVID-19, right?
Right.
So I was lucky to get there, but hopefully getting over soon so that you can come here to visit us at the
I hope so. Yeah. It's been tough. You know, I think that maybe once they get the vaccine distributed to the younger kids, and then this next wave goes through, hopefully it'll start winding down.
Yeah, that's correct. Yeah.
Stefan, I heard everything's back to normal in Germany. Like everyone's just kind of doing their thing. Is that true?
I mean, Germany still has mostly like mask requirement and things like that, but at least the labs and everything around there is kind of back to normal.
So do you wear masks in the lab?
In the lab, like in Munich, we weren't allowed in the lab for like chemical safety reasons. So as soon as you had like any chemicals working, then you weren't allowed. So there we had to keep strict distance regulation. But like in all the hallways, all the offices and stuff, we had it for a long time.
Okay, but now you don't.
In the Netherlands, like where I'm now, we only have it like really in the corridors outside of the labs that you're supposed to wear the mask.
How have you been, Chu?
Hi, Chris. Sorry. Yeah, long time no see. Haven't seen you for a while.
Yeah. How have you been?
Yes, this is great. Yeah, has been going on well, pretty well here at Beijing. Yeah. How are you?
Not bad. Not bad.
So you're in La Jolla now or in Florida?
Yeah, it's complicated right now. Right now I'm in Florida, but probably soon to be La Jolla.
I see. Yeah.
Stefan, how's parenthood?
At the moment, I'm here alone because my wife and my child, they will be traveling to the Netherlands only end of the week.
Sorry, where are you at right now? Have you moved?
I'm in Leiden, Netherlands, at the Leiden Institute of Chemistry. So really recently moved. So I started like my position here last week.
Congrats. Yeah, congrats again.
Are you located in Leiden right now, Stefan?
Yes, I am in Leiden, like
you already
starting
moved,
in September. Yeah.
Oh, cool. Okay.
Yeah. So Mario is actually like down the hall from me.
Got it. Nice.
Yeah, but still a lot of.
Do you have like raised ceilings in Leiden that count for you and the other giants?
That would make sense, yeah. Especially as it's a quite new building, so the others were already here, so they knew before they built it.
Yeah, they can anticipate the disproportionate size of the chemistry professors.
Another one of those.
Good to see you. Very nice talk. Really enjoyed it.
Hey. Wow, you look very European with that beard.
Yeah, it's after the lockdown.
Yeah, but it's not like, it's very well put together. It's not like some scruffy beard like Hacker.
Oh, thank you.
Like me.
You look much more erudite and polished. So how you been, Stefan?
Good. Yes, everything is fine. Thank you. Yeah. We also moved into a new building, so you still remember maybe our old one? No, we just recently relocated, which is very exciting. It's actually very nice here.
Oh, that's good. So you it's in the same general region or no?
Yeah, no, it's the same campus. It's just a new building next to the old one. So many things happened since you visited us last time. So might be good for a new visit at some point.
Yeah, we got to get rid of this virus. And I miss Munich, as you know. I think that's like the coolest city in Europe. So it's tough. Because I haven't been there in a long time. So
yeah. I remember last time we went to the beer garden in, I think, Chinesischer Turm, Chinese Tower.
Yeah. I'm gonna have to disagree with the coolest city though, but.
Sorry. Yeah. Sorry about that. My favorite Munich story, Stefan, from that trip though is still, Jacob was about, 6 or something, and we were in this. I can't remember. I don't think it was the English Garden. It was some other park where there's this famous statue of some athlete, and I can't remember what the. You know what I'm talking about? Like
it's Michael Jackson.
Is it?
Yeah. So I mean, it's not Michael Jackson, the statue, but I think this is in front of the hotel, the very Bavarischer Hof, and there many people are remembering Michael Jackson due to his visit here in Munich. Maybe that's the one you're talking about.
Yeah, but right next to this statue of this athlete, Jacob's like jumping over this chain, right? And we keep telling him not to jump over the chain, and he's trying to show how athletic he is, and then he tripped and fell flat in his face, right? And that was 5 or 6 year old crying for 2 hours walking through the streets of Munich after he face planted, jumping over a chain next to a robust German athlete. That was my memory of Munich. But
okay, great guys. I think
Yeah.
Good to do some catching up, but I think we should continue. So I will hand over to Monique now for the next session. Stop my screen sharing.
Yeah, thanks, Stefan. I was just about to interrupt the discussion indeed. So indeed, it's time to open up our second session of today and introduce our next speaker. So that will be Amy Weeks from the University of Wisconsin. So Amy, maybe you can already start your slide deck. So in the lab of Amy, by combining protein engineering, organic chemistry, and mass spectrometry, her lab studies post-translational modifications with spatial and temporal resolution to advance our understanding of PTMs and their biological function. And today she will tell us about her recent work mapping protein modifications across space and time in living cells. So Amy, the stage is yours. You need to unmute yourself.
Okay, sorry about that. And can we see the right version of my slides?
Yes, that looks fine.
Okay, thank you so much for the introduction and for the invitation. I'm really thrilled to be here with you today. And I'd like to tell you about some of the work that we've been doing in my lab, working on developing chemoenzymatic tools for mapping post-translational modifications with spatial and temporal resolution in living cells. So one thing that's become really clear since the completion of the Human Genome Project is that the human proteome is vastly more complex than the human genome. So while there are about 20,000 human genes, there are at least 1 million human protein variants or proteoforms. And while some of this protein level diversity arises at the level of alternative splicing, the vast majority of variation comes from regulated chemical modification of amino acid side chains or changes in peptide bond connectivity, so post-translational modifications. And PTMs regulate nearly all biological processes, and when they're dysregulated, it often leads to disease. But despite their pervasiveness and importance in biology, only a few % of PTMs have known functions, and these have been determined mostly by low-throughput experiments on individual proteins. And from these low-throughput experiments, we know that one important function of PTMs is to direct dynamic changes in the subcellular localization of signaling proteins, and we heard about an example of this from Professor Cravat this morning. And this can serve to regulate cell signaling on 2 levels. So at the level of signaling enzymes, subcellular localization controls access to substrates and binding partners. And at the substrate level, introduction of a post-translational modification might trigger relocalization of the substrate, which impacts its ability to perform downstream functions. So for example, extracellular stimuli can trigger assembly of signaling complexes at the plasma membrane upon receptor phosphorylation. And latent cytoplasmic transcription factors can undergo nuclear translocation following proteolytic cleavage. And proteolysis and phosphorylation can also activate and trigger relocalization of kinases. And upon reaching their destinations, all of these signaling proteins can perform downstream functions, which might include triggering further signaling cascades or executing transcriptional programs or orchestrating morphological changes to the cell. So what I hope these examples that I've shown here illustrate is that the dynamics of spatial organization are an essential feature of eukaryotic cellular signaling, and that if we had the ability to map the dynamics of PTMs with subcellular spatial resolution on a proteome-wide scale, this would be really transformative for understanding PTM function, and it would open up opportunities for both fundamental biological discovery and identification of new therapeutic targets. And the current sort of state of the art for studying PTMs on a proteome-wide scale is using mass spectrometry-based proteomics. But spatially resolved analysis of PTMs remains significantly more challenging than measuring bulk protein abundance or even bulk PTM abundance. And this is because PTMs are often present at low stoichiometry, so they have to be enriched to remove high background from unmodified proteins. And this reduces sample complexity and increases sampling depth. And the current methods rely on, for PTM enrichment, rely on the use of affinity reagents like antibodies or immobilized metals after cell lysis. So this leads to a loss of spatial information. And another challenge is that certain subcellular compartments can't be purified using standard biochemical fractionation approaches. And while genetically targetable proximity labeling enzymes like Ip6 and BioID and TurboID have really transformed our understanding of subcellular subproteomes, these enzymes don't enable specific capture of PTM sites. So new technologies for spatially resolved mapping of PTMs are really needed, and that's the challenge that my lab aims to address. So our overarching goal is to develop new methods for PTM proteomics with subcellular spatial and temporal resolution by engineering genetically targetable PTM selective proximity labeling enzymes. And these will enable us to modify PTM sites with biotin, for example, and then enrich them and capture these PTM sites with subcellular spatial resolution. And we chose an enzymatic approach for this based on some of the key advantages of enzymes, which include their ability to be targeted almost anywhere in the cell by genetic fusion to targeting domains like peptides or proteins that are localized to particular locations. And because the vast repertoire of natural enzymes includes many enzymes that target PTM sites for covalent modification as their natural activity. So our goal for enzyme engineering is to identify an enzyme that targets the PTM of interest, and then to engineer a variant that can modify the PTM site without regard to what the sequence context is. And we'd like to be able to leverage that activity to modify the PTM site with either biotin or a bioorthogonal probe. And once we've engineered an enzyme that has these properties, we can express it in a spatially resolved manner by fusing it to a targeting domain that will send it to a particular location within the cell. And then using that tool, we can induce spatially resolved biotinylation of PTM sites. And then we can enrich these post-translationally modified peptides and sequence them by LC-MS/MS to get a spatially resolved map of where PTMs resided within the cell. And my lab is initially focusing on two really pervasive PTMs, which are proteolysis and phosphorylation. And these both play really critical roles in biological signaling pathways. And today I'd like to share with you some of the work that we've done to apply this PTM capture approach to study proteolysis, which is one of the most common PTMs in human cells. And this is work that began during my postdoc with Jim Wells at UCSF, and it's now continuing in my own lab at the University of Wisconsin. So proteolysis is a really key post-translational modification, and it regulates a wide array of biological processes. So just to give a few examples, proteolysis is important in viral infection and viral propagation, replication, and release. It's also important in cancer progression and in the tumor microenvironment, where it can mediate cell-cell communication. Restricted activation of proteases, so spatially localized protease activity, is also really important in neural development and is dysregulated in neurodegeneration. And finally, as a last example, proteolysis is really important in organismal development, where activation of proteases in specific cells that are no longer needed can eliminate those cells in processes like digit individualization. So I think what these examples show is that the biology of proteolytic signaling can be really complex and can produce these organism-level phenotypes. But these phenotypes are built up on the molecular level from specific proteolytic cleavages that can impact enzyme function, protein localization and trafficking, and signaling at the cell surface, to name a few examples. So to understand the role of proteolysis in regulating biology, we need to know the exact site of proteolytic cleavage to get an idea of how cleavage will impact protein function. And although the biology of these proteolytic networks can be really complex, the chemical consequences are universal. So proteolytic cleavage always produces an unblocked N-terminal alpha amine. So if we could take advantage of the unique chemistry of this N-terminal alpha amine to capture it, then we could study proteolytic signaling on a systems level with single amino acid resolution for the cleavage site. And in the Wells lab, before I got there, they had developed an N-terminal peptide ligase called Cytosolic to modify unblocked N-termini. And this enzyme was engineered from the cytolysine protease scaffold by introduction of a cysteine in place of the serine nucleophile that we would see in the protease, and then introduction of a second proline to alanine mutation to better accommodate this larger cysteine nucleophile. And together, these two mutations abrogate protease activity, but they still allow for formation of this thioacyl enzyme intermediate. And this thioester is much more reactive toward amines than toward water. So in the presence of an N-terminal alpha amine from a peptide or protein, peptide bond formation actually dominates over hydrolysis. And this reaction occurs with absolute chemoselectivity for N-terminal alpha amines over lysine epsilon amines. So it's very site-specific. And this has been applied in a variety of contexts, but one that I'll mention today is that it's been used for studying proteolytic cleavage in this workflow called the N-Terminomics Workflow. And N-Terminomics refers to global capture of N-termini from a biological sample. So the way that this works is that we can start with a human cell line in which, for example, we've induced apoptosis. And then following cell lysis, we can get a protein extract and then use cytoligase to label all of the unblocked N-termini in that protein extract. And then we can enrich these labeled proteins on immobilized avidin and do an on-bead trypsin digestion to isolate just the N-terminal peptides from those proteins, which will allow us to sequence the proteolytic cleavage site. And then we can selectively elute these N-terminal peptides and sequence them by LC-MS/MS. And even without any filtering, you can see that we can recover the DEVD motif that's characteristic of the caspases that operate in apoptosis from an experiment like this. So when I started my postdoc, I was really excited about applying this type of approach in live cells since the previous work had only been done in lysate. And one of the areas where we were really excited to apply cytoligase was at the cell surface, because cell surface proteins are often underrepresented in other types of N-terminomic data sets, even though cell surface proteolysis is really important in cell-cell communication and has big implications for human health and human disease. So to target cell surface proteolysis with cytoligase, we fuse the enzyme to a transmembrane domain of the PDGF receptor beta chain to localize it to the extracellular side of the plasma membrane. And if we stain for cytoligase expression, shown in red here, we can see that cytoligase is indeed localized to the plasma membrane. And if we then look for biotinylation activity in green, we can see that the activity is co-localized with cytoligase. And this depends on the presence of the catalytic cysteine. So if we express a catalytically inactive mutant, we don't see any biotinylation activity. So then we can use this cell line expressing cytoligase in a modified N-Terminomics workflow in which we add a biotinylated cytoligase substrate to the cells expressing cytoligase on their surface and do the labeling in the context of live cells. And after biotinylation, these N-termini are covalently modified, so all of the spatial information is preserved. So we can then lyse the cells and do our normal enrichment workflow, and then elute these biotinylated N-termini that are derived from the cell surface and sequence them by LC-MS/MS. And what we find when we apply this new technology is that if we compare it to N-terminomics data sets that were generated by adding soluble cytoligase to a cell lysate, we see in that context, most of the N-termini that we identify are from intracellular proteins or found in intracellular compartments. Whereas if we use cell surface tethered cytoligase, we get about a 5-fold increase in cell membrane and secreted proteins compared to lysate. And we see a corresponding decrease in intracellular proteins when cytoligase is tethered to the membrane. And if we look at where topologically these N-terminal peptides are derived from, either in the context of soluble cytoligase and cell lysate or the cell surface tethered variant in live cells, we can see that in cell lysate, there is usually no topological annotation for the N-terminal peptides that we isolate. And this is because these peptides are derived from proteins that have no orientation. So they're just completely contained within one compartment and not tethered to a membrane. On the other hand, if we look at the data set from cell surface tethered cytoligase, about half of the peptides that we identified are actually derived from annotated extracellular domains. And these are just annotations that come from UniProt. If we look at the peptides that aren't annotated as being extracellular, they actually have a lot of features that we can identify that tell us that the peptides are probably extracellular, even though they didn't actually have that annotation. So many of them come after signal peptide cleavage sites or between a signal peptide and a transmembrane domain, indicating that they'd be on the extracellular surface of cells, or between signal peptides and a GPI anchor, or have some other features that would suggest that they're on the extracellular surface. So with this high level of cell surface selectivity, we've been able to use this as a tool for quantitative proteomics to look at how proteolytic shedding at the cell surface is connected to kinase signaling. And now we're working on expressing these constructs in mice and worms to use them to study proteolytic signaling in whole animals. So the cytoligase transmembrane construct is a really robust and versatile tool for studying proteolysis at the cell surface. But at the beginning of the talk, I told you that we wanted to study PTMs like proteolysis inside live cells and in various different locations and compartments, not just at the cell surface. But subtle ligase requires a cell-impermeable peptide ester as its substrate, so it's not really very well suited to capture proteolytic events inside living cells. So in the next few minutes, I'd like to tell you about one of the approaches that my lab is taking to overcome this challenge. So in talking about subtle ligase, I focused on capturing the neo-N-termini generated by proteolytic cleavage. But of course, proteolytic cleavage also generates a neo-C-terminus. And capturing either the N-terminus or the C-terminus would tell us where the cleavage site is after proteolysis. And we could also, if we had a tool to capture C-termini, we could use this to study proteolysis on a systems level. So one new direction that my lab is going in is to develop enzymatic tools that enable spatially resolved capture of neo-C-termini inside living cells. And one reason that we chose to go in this direction is that although C-terminal carboxylate groups are pretty unreactive on the scale of compared to N-terminal alpha amines, there's actually a large and diverse superfamily of enzymes, the Thiaf Superfamily, that can activate protein and peptide C-termini using ATP, and that's a substrate that's readily available inside living cells. So we thought that this Thiaf superfamily would be a good starting point for protein engineering to develop a C-terminal modifying enzyme. So the Thiaf superfamily includes homologs across all kingdoms of life, and there are a few notable families, including the Thiaf-MoV family. These enzymes are involved in cofactor biosynthesis. The E1 ubiquitin ligases, which are involved in ubiquitin transfer, and then the MCCB enzymes, which are involved in antibiotic biosynthesis. And common to all of these enzyme families is the formation of this reactive mixed anhydride intermediate that results from adenylation of the C-terminus with ATP. And then in all of these families, this intermediate is captured with a nucleophile in downstream reactions. So we decided to choose the MCCB family as a starting point for protein engineering because unlike some of these other families, MCCB acts on peptide substrate, so it doesn't rely on protein-protein interactions for substrate recognition. So in its native context, MCCB acts on peptides that have a C-terminal asparagine residue. And the proposed mechanism involves adenylation of the C-terminal carboxylate of this N-terminal or C-terminal asparagine side chain to generate a mixed anhydride intermediate. And this intermediate is then captured by the carboxamide of the asparagine side chain to generate a succinimide intermediate. And this intermediate can be adenylated a second time to generate this stable phosphoramidate product. In its native context, this product is involved in microbial competition. So what we're working toward with MCCB is to engineer it for promiscuous C-terminal adenylation activity so that any C-terminal residue would be accepted for adenylation. And then our strategy is to capture this mixed anhydride intermediate with an exogenous nucleophile since there wouldn't necessarily be a side chain nucleophile available. And if this were biotinylated or labeled with a bioorthogonal group, we could then capture the C-terminal peptides and analyze them quantitatively using LC-MS-MS. And this project has been spearheaded by Claire Fraser, who's a talented graduate student in my lab. And Claire has expressed and purified MCCB, and she's reconstituted its native activity with the C-terminal asparagine peptide so we can detect the stable phosphoramidate product that's formed. So our next question was, in the absence of an asparagine side chain, can the C-terminus of the peptide get adenylated to form the mixed anhydride intermediate, which we would expect to be very unstable in water? So to address this question, Claire synthesized 19 mutants of the native substrate where the C-terminal residue was varied to all the other amino acids. And because we expect this mixed anhydride to be unstable in water, we detected adenylation activity by looking for pyrophosphate using an enzyme coupled assay. And what Claire found is that although this stable phosphoramidate product is only formed when the asparagine side chain is present as shown in the left column here, in many other peptide sequences with different C-terminal residues, the adenylate is actually formed, so we see production of pyrophosphate. So MCCB is fairly promiscuous in terms of this first step. So we wanted to really convince ourselves that the adenylated intermediate was forming in the context of these peptides that don't have a C-terminal asparagine, and also to test our proposed nucleophilic capture strategy. So Clara has screened a variety of hydrazine and alkoxyamine nucleophiles to test whether we can trap the mixed anhydride intermediate that we believe is forming in this context. So here I'm showing some experiments that Clara did in the context of the peptide with a C-terminal glycine residue. And what she found is that when we do this reaction in the presence of ethoxyamine, we can actually capture this adenylated intermediate. So we see a nice shift by HPLC and a corresponding mass shift corresponding to the ethoxyamine modified peptide. And this is enzyme dependent, so we only see it in the presence of MCCB and not in the absence of MCCB, even when ethoxyamine is present. So this provides additional evidence that the mixed anhydride intermediate is formed for this substrate, and also that it's amenable to capture with an exogenously added nucleophile. So the next thing that we'd like to do is to optimize MCCB for promiscuous C-terminal adenylation. And to do this, we're taking a structure-guided engineering approach in which we first perform alanine scanning on all of these residues that are shown as gray spheres here, so those that are interacting with the peptide based on structural information that's available. And Clara has made all of these mutants, and here I'm just showing a subset of her data in which she screened them against her 20-member MCCB substrate library with different C-terminal residues. And using this assay, we've identified several alanine variants, including Y258A and S212A, that are even more promiscuous than the wild-type enzyme for different C-terminal residues. So going forward, we're targeting these sites for saturation mutagenesis to try to further enhance the promiscuity of MCCB. And we'd also like to understand what residues beyond just the very C-terminal position influence substrate specificity. So we've addressed this a little bit by synthesizing different peptide substrates, but we also wanted to develop approaches that would allow us to screen a more highly multiplex panel of substrates that goes beyond just our 20-member synthetic peptide library. So one approach that we're developing for this is to use C-terminally diverse proteome-derived peptide libraries. And in this workflow, what we do is start with a protein extract from E. coli and then digest it with a protease that generates C-terminal diversity. And we can use this as a pool of substrates for MCCB in the presence of a nucleophile to capture the adenylated products. And then we can enrich and sequence these by LC-MS to read out the enzyme specificity. And in preliminary experiments, we've used this C-terminally diverse proteome-derived peptide library and captured the adenylated peptides. And what we find is that it appears that the C-terminal residue is the most important for MCCB specificity. So we're really optimistic about our ability to engineer MCCB for promiscuous C-terminal modification. And we're planning to use this assay as an engineering platform to do that. So ultimately, what we plan to do with this tool, once we can engineer it, is to genetically fuse MCCB to various targeting domains for spatially resolved mapping of proteolysis in living cells. And more generally, we think our enzymatic approach for PTM capture can be generalized to any PTM for which a modification enzyme can be identified. So other projects in the lab are focused on applying this type of approach for mapping phosphoserine, phosphothreonine, and phosphotyrosine with spatial and temporal resolution in live cells. So with that, I'd like to thank all of the members of my lab, particularly Clara Fraser, who spearheaded the MCCB project, and also Jim Wells, my postdoc advisor, and Jamie Burns and Irene Louie, who also contributed to the Subtle Ligase TM project. And thanks for your attention, and I'm happy to take questions.
Great talk, thanks. So if you have questions, please put them in the Q&A. So I was actually wondering, since you are now using your engineered protein, does it also interfere with the natural processes where indeed the adenylation plays a role?
So we actually haven't tested this in live cells yet, and we think that what we're hoping is that it won't interfere with natural adenylation processes or other natural processes that depend on the C-terminus because the intermediate is so unstable. So if we use our nucleophiles and only capture a small percentage of it, of C-terminal peptide that we can enrich, since mass spec is very sensitive, we think we'll still be able to read out those cleavage events without impacting the biology too much.
Okay, thanks. Then we have a question from Steven Sieber. So he wonders if you consider other strong nucleophiles for mixed anhydride capture, such as hydrazine derivatives.
Yes, so we have tested hydrazine nucleophiles, and they also work. One issue that we've run into and haven't quite worked out yet is that when we do our tandem mass spec, there seems to be some issue where even though we can detect them by LCMS and we can see the shift on the HPLC when we capture these hydrazine nucleophiles, we can't really identify them in a tandem MS experiment. And we think that that might have something to do with how they fragment in the MS2, and we haven't quite figured that out yet.
Okay, thanks. Then another question, are there any PTMs beyond protease cleavage that you are specifically interested in exploring with your technology?
Yeah, so beyond protease cleavage, in my lab, we're working on phosphorylation. So phosphorylation is probably one of the most studied and most widely occurring PTMs in cells, but we think adding a level of spatial resolution would be really useful for that. And we've identified a couple of enzyme families that will enable us to modify either phosphoserine and phosphothreonine or phosphotyrosine. And we're still working on those. And I guess those stories are maybe at a little bit earlier stage of development, so I didn't talk about them today.
Okay, thanks. So do you think that also your technology will allow to look at the crosstalk between PTMs?
So I hope that it will. I guess basically the goal is if, say, a year or two from now, we have tools that enable us to map both proteolysis and phosphorylation, I think that would be really exciting to combine those and look at crosstalk, because in apoptosis, for example, we already know that this crosstalk between proteolysis events and phosphorylation events is really important. And I think no one has systematically mapped that with spatial resolution. So we are very excited to try to approach that problem with these tools.
Okay. And then we have a question from Mario. He would like to know if you identify new substrates for the Cas bases with this new technology.
So it is our hope that we'll identify new substrates, and actually we haven't done that experiment yet, but it's sort of in the pipeline. And we think one reason that having a new tool will be really useful for expanding coverage of Cas-based substrates is that many of the N-Terminomics methods that actually have single amino acid resolution for the cleavage site rely on identifying one peptide per protein in a mass spec experiment. So if you have a cleavage site that doesn't fall into a peptide that is identifiable by mass spec, then you would miss it. So we're hoping that by having an orthogonal tool, we can catch more of those and expand what we know.
Okay, thanks again for your great talk. And then we'll move to our next speaker. So the fourth talk of today will be given by Chu Wang from Peking University, China. And his research group studies protein function by the development of chemical and computational proteomic methods. And today he will talk about his group work on identifying new metalloproteins in proteomes. And his project is called Profiling Metalloproteomes by Chemical and Computational Proteomics. So please, the floor is yours.
Thank you for the kind introduction. I assume everyone can hear me and see the slides properly?
Yes.
Okay. Thanks for the introduction, and also thanks, Stephen, for the kind invitation. It's a great opportunity to be in this symposium and also catch up with my postdoc advisor, Dr. Ben Cravat, and also other friends from the Cravat lab. So today, I would like to share some of my work from my laboratory on profiling metalloproteomes by chemical and computational proteomics. So as we all know that we enter the post-genomic area, and also with high development of the high-resolution mass spectrometry, now we can have access to the rich information of the human proteome. And several years ago, they established the complete map of the human proteome. So that's also leave us with a daunting question on what exactly the function of those proteins are. So as we know that there's a different type of functional sites in the proteomes. Proteins can do catalysis, and they undergo different type of post-translational modifications, and they can also their activity can also be regulated by different type of ligand endogenous metabolites, and also protein forms protein-protein interactions to perform their function. So if we can have a high throughput ways to identify those type of functional sites, that will help us to get more information on what the function of those proteins. So that's when I came back to Peking University about 8 years ago. My laboratory was set up to combine the chemical proteomics together with in silico computational proteomics, trying to discover different type of functional sites in the proteomes, and also to developing chemical biology tools to regulate such functional sites. So to give you a few brief example briefly, so in one of the work we developing photo cross-linking probes based on a traditional Chinese medicine natural product called Backlin, which has very unique activities to reduce reduce the lipid deposition in the livers, so that we use this chemical photo affinity probe to capture its target proteins, and among them we find it actually can activate a specific enzyme in the metabolic pathways, and then to have this specific biological activities. And in another work on regarding the PTMs in collaboration with my colleague Professor Xun Chen's lab at Peking University, so we use one of the unnatural sugar probe they developed, which has this unique cysteine reactivities, and then using a competitive ABPP platform, as Ben talked in early in his talk, and then we are looking at the specific modification by the this metabolite called the itaconic acid, which has unique anti-inflammatory and also antibacterial activities. So we find doubts that the itaconic can actually modify cysteines in several key enzymes in the glycolysis pathways, and mediate this anti-inflammatory activities. And from the computational side, this is another collaboration between my lab and Professor Peng Chen's lab, also at Peking University in our department. So we use a computational design trying to pinpoint the specific residue positions where we can insert unnatural caged amino acids, so that it can block the enzyme activities, and after the photo decaging, it will remove the protecting group and then unleash the activity of this enzyme in the living cell. So we demonstrate this could be with a computational design, it could be applied to a variety of different enzymes, and then having their activities directly activated in the living cells. But today I want to share with you two works which are ongoing in our lab right now regarding on the metalloproteomes. So the first one is led by this talented graduate student Xin Zheng, and then a postdoc Dr. Yuan Liu, on using chemical proteomics to profile the metal-binding proteins. So I think I don't need to emphasize the importance of the metal-binding proteins in biology, because they perform different type of functional roles, such as the transcription, catalysis, signal transduction, and the storage. And like I said, been estimated that about 1/3 of the proteomes are actually metal-binding proteins. So currently in the field, there are several classical methods have been developed to discover metal-binding proteins in the proteome, including using the ICP with LC mass spectrometries, and to identify metal-binding proteins. And you can also using the immobilized metal affinity chromatography to specifically enrich out some metal-binding proteins for mass spec identifications. And also labs are also taking advantage of the labeling of the specific residue sites in the proteome by IBPP technologies, so that you can use this competitive version of that to looking at the zinc binding proteins specifically. But we thought maybe there's other physical properties of metalloproteins that can be exploited by us to develop new method to looking for metal binding proteins. So we were inspired by this classical paper published in biochemistry almost 25 years ago, which shows that actually when a mito is removed from its target proteins, they actually can affect the thermal stability of these proteins. And on this specific enzyme called arginase, if you remove the metal, it's getting destabilized. And also, if you mutated a specific metal-chelating residue in those enzymes, you also see the similar effect. So that's sort of give us the inspiration, because this has been a very popular technique called the thermal stability profiling technology, also in the short for TPP, has been developed for the drug size identification or the drug target deconvolution technologies, where you can basically using the multiplex mass spectrometry quantitative proteomics such as TMT to measure out the thermal stability of proteins across the proteomes upon the binding of a specific compound. So we thought maybe we can combine these two technologies together and then create this technology called the metal TPP, which the idea is we can use a common metal chelator such as the EDTA to perturb the proteome, and then use the regular TPP mass spectrometry to measure the thermal stability shift of the certain proteins. So we thought this could be a very simple assay, because you just need to treat the proteome with the EDTA, and also it will be general for discovery different type of metal binding proteins, since EDTA is a broad scope metal chelators, and then this allow us to maybe discover new metal binding proteins which haven't been annotated before. So we first trying to demonstrate this concept on a purified protein. So we use this zinc binding protein DHSO as a positive control, and you can see that if you adding treated protein with EDTA, it will generally destabilize the proteins, and also you can sort of measure the intensity on the gray signal on the Western blot, and you can sort of see that there's a melting curve shift. And this thing actually works for other type of metal proteins, such as magnesium binding proteins of ENO1. And then for the non-metal binding proteins, such as GAPDH, you don't see such an effect there. So we then just went back to the proteomic profiling, and in the proteome, we just treat the proteome with the EDTA, and then using the TPP to measure globally the melting temperature, melting curve shifts. And among them, we quantify about almost 3,000 proteins, and along those proteins, a large portion of those with destabilized shift is actually annotated metal binding proteins previously, and also all those red points are actually some new previously unannotated metal binding proteins. So actually, if you compare the proteins with reduced thermal stability in our assay, for those known metal binding proteins, the metal type distribution is quite similar to the ones known in the UniProt database. Again, supporting that there's actually general scope for the EDTA to extract the metals in different type of proteins. And if you look at the list of the proteins with large melting temperature shift, the delta TM, large delta TM, there's quite a few proteins which don't have really like a functional annotation before. And among them, we focus on this specific protein. This actually is an enzyme called the GFPT2. It's a glutamine fructose 6-phosphate aminotransferases. And this protein actually performs an important role in this hexane sugar biosynthesis. It will convert fructose 6-phosphate to this glucosamine 6-phosphate. And it's actually have a conserved ortholog in the E. coli. The enzyme is called the GLNS, perform the similar functions. So we start trying to do some biochemical validation using the GLMS as an analog. And between these two proteins, two orthologs have a very high sequence similarity to each other. And actually, this protein is also have two domains. On the N-terminal domain is glutamine binding proteins, and on the C-terminal domain is this isomerase binds to fructose 6-phosphate. So we first purify the GR mass, and then we observe that specifically only zinc can actually stabilize the structure of these proteins, as shown in the thermal melting curve here. And we also measure the activity of this enzyme using the biochemical assays, and then we find that actually zinc can actually inhibit the enzyme activities. And but if you after that you treat it with the EDTA to extract the zinc, you can sort of reverse back the activity there. And this effect also seems to be zinc specific. And in collaboration with my colleague, Professor Xiao Junyu at Peking University, who is a crystallographer, they help us to solve the structure of the GMS C-terminal domain. Actually, across, we solved the structure as a dimer, and across on the dimer interface, we saw 3 like zinc ion there. And if you look at the zoom-in view, in 2 of them is kind of symmetric, is basically coordinated by one of the cysteine and another histine across the interface. And then there's another one zinc binding site which has coordinated by 3 histine there. And if you measure the zinc binding stoichiometry using ICP mass, we actually see that the stoichiometry is actually about 1 to 1.2. So sort of indicating that there may be one zinc binding site is not fully occupied. So as I mentioned, that this is a 2-domain protein enzymes, and on the C-terminal domain, this observed the zinc binding residue cysteine 301 and the histine 505 is actually close to the fructose 6-phosphate binding pocket. And based on these observations, if we using 2 different substrate to treat the enzyme, we can only observe the competition on zinc binding if we use the fructose 6-phosphate to compete the zinc binding as consistent as we observed in the structures. So to summarize this part, we developed this quantitative chemical proteomic platform called the MetalTPP based on the thermostatic profiling, and then to identify around 100 potential metal binding proteins. And we actually validate one of the cases, GLMS, as a zinc binding proteins. And we also solve the crystal structure to show that it's indeed a zinc binding proteins. But for the future plan, we definitely want to investigate what is the downstream physiological role of zinc on hexamine biosynthesis, and also in terms of the technology development, whether we can achieve metal type specific profiling and also map the specific, actually the exact metal binding site. And for the second part of my talk, I want to switch gear to the another work using a computational method trying to predicting metal binding proteins, and in this case, specific metal binding sites in the proteomes. And we actually use the sequence coevolution information to help us to make such predictions. And then this project was led by also 2 talented graduate students, Dr. Hao Bo-Wang, who graduated a couple of years ago, and then Ms. Yao Cheng, who is in his 5th year right now. So what is sequence coevolution? So as you have a multiple sequence alignment of a specific protein, you always observe this conserved position highlighted in green, and also you see a lot of positions are highly variable. But there's also, if you look at the analysis more closely, there's actually other type of positions where such, for example, there's a pair of residues who actually changes together across different orthologs, so that means they actually co-evolve together. So actually, coevolution can be now easily computed by different methods and based on the multi-sequence alignment information, and then you can sort of draw display such analysis using by this is called a contact map, which from the N-terminal and C-terminal in this matrix, and then each dot here representing whether these two residues in the proteins are co-evolved together. And if you blow up this specific dot, you will see the composition of different amino acid in the evolution history between those two positions. So actually, the sequence coevolution has been used in the structure prediction field to predict accurately the protein structures and also protein interactions. This is based on the assumption that if two residues co-evolve together along the evolutionary history, they probably physically interact with each other, or like they are functionally conserved. So actually, people have done such analysis before on individual proteins. They also observed that in both the enzyme active site and also protein ligand binding site, as well as metal binding site, they observed such coevolution before. So that's what inspired us to developing a global computational proteomic method to see if we can harness such information to help us to predict the metal binding sites in the proteins. So we first constructing a benchmark of the metal binding proteins from the protein data banks, and then finally got about 500 metal binding proteins. And among those proteins, we did an initial analysis which shows that actually a majority of the metal chelating residues are cysteines, histidines, and two acidic residues as we expected. So we basically, for the remaining of the talk, we focus on these four residues, shorted for CHED. And then we divided the metal binding chelating residues in those metal binding proteins into two data sets. One is a positive data set, which means they are metal binding residues, which also shows a co-evolution, co-evolved CHED pairs. And then there are also a large number of the non-metal binding co-evolved CHED pairs used as negative negative data set for training. And then when we did a co-evolution analysis, we actually see that for a lot of CHED pairs, such as the cysteine and histine in the metal binding site, they actually have a higher percentage of the co-evolution signals than the metal binding sites in the random pairs, as shown here. So, which means that the metal binding residues are indeed more co-evolved than the rest part of the proteins. So also, if you're looking at the specific, the so-called frequency matrices showing the co-evolution history, you immediately can tell that metal binding residue pairs have a very unique look, which is distinguishable from the non-metal binding residue pairs. So this type of the image problem can be just readily fed into the machine learning algorithms, such as convolutional neural networks, and you can just feed it the frequency matrix calculated from the co-evolution signals, and then having the algorithm to help you tell whether based on these pictures or based on this feature, whether this is a metal binding pair or non-metal binding pair. So we basically starting from the multi-sequence alignment and then created a contact map. And then from there, we extracted the CHED residues and then looking at this their specific frequency matrix and then using machine learning model to predict whether this is metal binding or non-metal binding. And also, there's another features you can take advantage, you can use for the prediction. As we mentioned, for the first step, you're only looking at a pair of residue, whether they are metal binding or not. But as we know that a lot of metal binding sites, they actually are composed by multiple residues. And for example, like a zinc finger or iron sulfur cluster binding sites, there's like a cysteines, maybe 4 different residues, they all co-evolve together. So instead of having, in addition to having like a specific individual pair of the co-evolved metal binding residues, they will form a more higher order, like a co-evolved networks. So in this case, we further developing algorithm based on the graph theory, trying to assemble those individual metal binding pairs into a more complicated network here. So if we can get such a completion of the network, it not only gives us more confidence that this is more likely to be a mito binding site, but also in some cases, you can actually, based on the topology of this network, you can go back to query against the mito binding networks observed in the protein data banks, and then to help you to infer the type of the mito binding information in these proteins. So we call this whole flow MitoNet. Again, we started from the multi-sequence alignment and using co-evolution analysis. Based on the machine learning model, we can predict each individual pair is more likely to be mito binding or not, and then we transform them back to the higher order networks. If we observe such networks, we have a higher confidence that this is more likely to be a mito binding site. And then maybe in some cases, we can also infer the mito binding, what the the information for the specific mito ions binding there. So we apply this method to the 4 prokaryotic bacterial species, and then among those, among almost 10,000 proteins, we identify a lot of potential mito binding pairs, and then from those there we predict about more than 4,000 potential mito binding sites, and then cluster them into this 1,200 mito binding clusters. And then this is actually dramatically increase the number of the currently annotated mito binding proteins in those bacterial proteomes. And then actually our prediction reach approximately about 1/3 of the overall proteome in those bacteria. And some of them actually have been, if we do an analysis on our predictions, half of them actually can be supported by the remote homologs observed in the protein data bank, and then there's also a large portion of them can be supported by some other annotations in the like UniProt or the PFAMs. So to give you a few examples, so this is a prediction made by the MitoNet on the top left corner here. This is actually predicted networks for ion sulfur binding cluster proteins. And here is actually, it's a very complicated network, but it corresponding to this trinuclear mito binding site. And on the bottom here is actually from this protein, we predicted 2 different type of the mito binding networks, and corresponding to 2 different cluster of mito binding copper binding sites there. And also there are cases where when we find the structure, there's actually no metal there, but based on the structure, it looks like it should have a metal there. Probably when people solve the structures, if they don't know if it's a mito binding proteins, they maybe the buffer they use will cause the mito to lose. And there's also cases where there's a missing densities, and also low resolution structures don't support mito binding. But later they solve the high resolution structures, it actually show that there is clear the mito binding site there. So among our predictions, there's a lot of proteins in those bacterial species are still uncharacterized proteins known as acrized proteins. And then we think to give a little bit information on whether they are mito binding could help other people to study their functions. And also there's a large number of the also potential ion sulfur cluster binding proteins predicted by our method. So we actually biochemically validated one of the examples is a protein enzyme called CITX from the E. coli, and our MitoNet predicted a very good four residue clusters, and looks like a zinc binding site. And then we actually purify this protein, and then if we use EDTA to treat the proteins, they actually can destabilize the protein significantly. And then we use the SCP mass to measure its mito bindings, and then we observe this strong zinc bindings. And if we mutate individual of the predicted mito chelating residues, it will abolish the zinc binding there. And then in collaboration with another crystallographer, Professor Xiaodong Su at Peking University, they help us to solve the structure. And then there's beautifully you can see that there's a zinc sitting there beautifully, very highly consistent with our predictions. So to summarize this part, we developed a machine learning method based on the sequence coevolution to predict metal binding sites in the proteomes. And then in addition to that, we also having the graph theory algorithm to assemble those metal binding pairs into a higher order networks to increase the confidence of our predictions. And from this work, we predict a large number of novel metal binding sites, and we validate one of them. And for the future plan, we want to extend it to the eukaryotic metalloproteins because this seems like more challenging. You need to get the multi-sequence alignment with the higher qualities. So with that, I want to conclude my talk and thank the people in my lab, and it's also the collaborators, Professor Xiaodong Su and Xiao Junyu, for help us to solve the structures. And this is a final agency to support this work. I would like to thank you for your attention.
Well, thank you for this very interesting talk. Already a few questions came in. And of course, please, if you have any questions, put them in the Q&A. So one of the questions is if there is a way to link the predicted binding to specific metals and also to specific enzymatic functions.
I'm sorry, I'm trying to catch up the first one, right?
No, sorry. No, it's
sorry,
via another platform. So you can now, of course, link the predicted binding to specific metals, but is it also possible to link those to specific enzymatic functions?
Yes. So the second part of the computational one, we can, based on our predictions, we can pinpoint the metal binding specific site. And in some cases, if the network is complete enough, the topology is complete enough, we can infer the type of metal binding there. Yeah, but we haven't really linked that specifically to different enzymatic activities. The two cases I show, the CITX one on the second part of my talk, we haven't measured its activity yet. But the first one, we do see that zinc has a inhibitory effect on the enzyme activities. Although it's hard to explain mechanistically what's happening, it could compete the substrate, the fructose 6-phosphate. But on the other hand, it's also as the interface. So that's still under investigation right now.
Okay, thank you. You also mentioned that you want to expand this technique to differentiate proteins binding to different metals. How do you envision to do that?
For the experimental part, I think currently our idea is because the EDTA is a broad scope metal chelators, and then we notice that people, especially in the bio-inorganic chemistry, they have developing the other like metal specific chelators. Some for zinc, some for maybe for copper, and some for iron. We thought maybe that can also give us some basically applying the similar technologies, and then we just applying the metal specific chelators that may help us to pinpoint the specific subpopulation of the metal binding proteins based on the metal binding type. That's one approach we thought about with the experimental techniques. For the computational one, I think currently the benchmark we collected is also we don't categorize them into different type of metal binding proteins. We basically take whatever the metal there and all the protein available. But if you train the model specifically with a subset of a protein, say zinc binding protein or iron sulfur cluster binding proteins, I think in that case you probably have a higher chance to get the information about what type of metal binds there.
Okay, thanks. So then there's also a question from Dorothea. She has a question about the first part. How do you think about the potential metal binding proteins you identified in which delta TM is positive?
Yeah, that's a great question. We sort of took a shortcut, at least for choosing the target for validation, because decreasing the delta TM is negative one. Those type of things are easy to interpret. I think the ones with the positive, there is another possibility because we treat the protein with the EDTA. It's actually a small molecule. It may the one possibility is that it will bind to some proteins and then just stabilize it. So that's one explanation. But I bet there may be other cases, because we have from the literature cases we found is when we remove the metals, it actually decreases, destabilizes the protein stabilities. But in terms of the small molecules, I think in TPP studies they see on both sides, on both directions. So I guess that if just let me to interpret that data, I think EDTA may have some other binding target in the proteomes. And also I need to clarify that this is sort of indirect, right? Because there could be also other cases where the EDTA have the effect of perturbation on metal binding proteins, but we read out the maybe it's interacting partners or maybe a subunit in a complex. So that's all other possibilities for that.
Okay, thanks again for your great talk. There's still a question open in the Q&A. Maybe you can enter that later during the break.
Yeah, because it's pretty late here in Beijing. I'll try to answer them as soon as possible. Yeah.
Okay, great. So then I would like to thank both speakers again, and we'll have another break and see you then in 15 minutes again for the next session.
Shall we start, Stefan?
Yes, please.
Okay, welcome back everyone to the third session of the Chem Bio Talks. My name is Mario van der Stelt from Leiden University, and it's my great pleasure to introduce you our next speaker, Dr. Yuna Yu. Please, could you share your screen? So Dr. Yu is the Statman investigator of the Chemical Biology Laboratory and head of the Chemical Immunology section of the National Cancer Institute in Maryland. And she obtained her PhD from the University of Kansas in the Department of Medicinal Chemistry under the mentorship of Professor David in 2015. She then joined the laboratory of Professor Matt Bojo at Stanford as a postdoctoral fellow. And her current goal is to discover novel chemical approaches and molecules that can address immunological problems in cancer therapy. And today she will tell us what we can learn from nature's electrophiles to design better covalent protein ligands. So please, Yuna, the screen is yours.
Thanks. First and foremost, I'd like to thank the organizers for the invitation. The project that I'm going to present today is still work in progress, but I'm really happy to share some exciting opportunities we are finding as we are making some progress. As you'll see in my talk, our work is really inspired by and also benefited from others in this Chem Bio community, including organizers and speakers of this very symposium. So my lab studies proteases involved in cancer and immunity at the interface between the protease biology and immunology. As we already heard from Amy, the proteolytic activity is involved in numerous biological processes through protein degradation, post-translational modification, and modulation of signaling pathways. Protease activity also is central to immune responses and can dictate the complex microenvironment. We are particularly interested in the roles that proteases play as instigators and regulators of immunomodulation and its relevance to disease progression in cancer and immune disorders. So our goal is to develop chemical strategies that specifically measure and perturb protease activity to dissect their functions, and also develop potential modulators that can be used as a diagnostic and also therapeutic tools. As one of the main approaches to study protease activity, we are taking this chemocentric approach that involves applying small molecule libraries directly to a complex biological system and identifying and validating protease targets that elicit the biological activity. In our study, we are looking for novel ligand protein interactions that can modulate immune responses. In particular, we are interested in studying covalent ligands. As we know, many immunomodulatory compounds can chemically modify nucleophilic residues in the immune-relevant proteins through covalent bond formation. And catalytic activity of various enzymes regulates dynamic immune cell signaling cascades. Demonstrated in the beautiful work by Professor Kravec and others, there are redox-sensitive and reactive cysteines that can be liganded in human immune cell signaling pathways. So by properly combining the features of the chemical reactivity and the molecular recognition, we thought we'll be able to find protein-ligand interactions that can present new opportunities. And this work is really spearheaded by 3 people in my group, David, Ray, and Robbie, with their collaborative efforts. First, we wanted to build our own library of compounds with the aim also to expand the scope of electrophilic chemotypes in warhead development, mainly with the mild reactivity that can confer high specificity. As a source for inspiration, we started looking at the electrophilic natural products. Personally, I have developed a great appreciation for electrophilic natural products as they were a critical starting point as a chemical probe for my postdoctoral work with Matt Bojo on proteasome and lipase inhibitors in Plasmodium, either in the covalent compound optimization for the target of interest or the identification of new targets with the complex natural product. And we found this compound acetylysine, which was originally isolated from the fermentation broth of the Streptomyces spices during the screens for the antibiotics. This compound has antibacterial, antiparasitic, and anti-cancer activities. And actually, it belongs to the non-canonical amino acid mimicking L-glutamine, thus inhibiting glutaminase activities. It contains this unique electrophilic moiety, the chloro-dihydrolysoxazole scaffold, that reacts with the nucleophilic residue by displacement of the chlorine atom and forming the covalent bond. We thought this warhead is interesting because it has been rarely studied as a covalent inhibitor, and it's got a mild chemical and solution reactivity. In fact, it doesn't readily react with the glutathione. As also proposed in this glutaminase inactivation mechanism, we wondered whether this protein binding associated activation and stabilization, like the presence of the oxyanion hole in the active site of an enzyme, is required for the covalent modification of this warhead. And also, we thought it might be possible that this warhead also can target not only cysteine, but also serine and lysine with the perturbed PK in the binding pocket. And actually, when Professor Sieber group investigated the biological targets of the activicin or the bromacivicin in bacteria and cancer cells, mainly using the probes functionalized by the alkyne handle on it, what they found was that these compounds have a very diverse and distinct pattern of the protein binding preferences depending on the small variations at the dihydrolysoxazole scaffold. And the set of dehydrogenase enzyme family was found to be a major target of the activicin. There has been a few bromoisoxazole containing inhibitors reported for targeting enzymes such as transglutaminase 2 and also the toxoplasma cysteine protease rhodosane. We first investigated whether simple BDHI probes conjugated with the tag could covalently modify the protein in human proteomes. We synthesized the BDHI probe with the Tamara or the PEG Tamara and used this probe to label the recombinant proteins such as transglutaminase 2 and aldehyde-dehydrogenase 1 that are known to react with the electrophilic moiety. Actually, the probes were able to label the proteins, as you can see here. And compared to broad spectrum cysteine reactive probe iodoacimide Tamara, we observed this BDHI probe could label proteins with somewhat less extent, but with some preference. Between the Tamara and the pegylated Tamara, the hydrophobic Tamara probes seem to serve better as a binding scaffold. Similarly, when BDHI probes used in the Zucca cell lysate, the probes could form the conjugates with the numerous proteins, as you can see here. Also, the probe conjugated to an alkyne functional group could modify proteins in the Zucca cell lysate through the covalent mechanism, although it required a relatively higher concentration of the protein, the probe, sorry. And based on these observations with the simple BDHI probes, we thought this warhead does not easily react with the nucleophilic residues unless they are correctly oriented near a nucleophilic side chain in the protein binding pocket. Thus, local, environmental factors and also the binding interactions with the proper scaffold might contribute to more to the proteome reactivity and selectivity. So we decided to install this BDHI warhead onto structurally diverse chemical scaffolds. After consideration of a few synthetic routes possible for the diversification, mainly utilizing 1,3 dipolar cycle addition using this precursor dibromoformaldoxine to offer the 5 substituted BDHI compounds, we first decided to explore the coupling reactions of this methionamine BDHI compound with the library of the carboxylic acid. And we are able to synthesize the first set of about 270 compounds. Next, we wanted to screen for their functional activity and also the protein reactivity, already explained by Professor Kravett. To quickly assess the protein reactivity and selectivity of the compound, we use this competitive gel-based protein profiling, where proteins are first treated with the inhibitors and then competitively labeled with the broad spectrum probe appended with the fluorophore. Probe conjugated proteins can be resolved on a gel, and the pretreatment with the inhibitor that blocks the site for that probe labeling results in the reduced fluorescent readout. Since we are interested in the immune signaling pathways, we decided to use Jurkat cells and the human T cells and compare those to other cell lyses such as HeLa and HEK cells. So the initial screening was done with the pool of 5 compounds, each at 500 micromolar concentration and 4-hour pre-incubation, followed by the IA tamer labeling for 1 hour. And the compound pools that show any sign of binding were deconvoluted to evaluate each individual compound. So these are the examples of the gel data with a few hit compounds, structures shown here. So as you can see here on the gel, compounds like the 151 and 258 showed some blockade of the site for the IA tamer labeling with some selectivity, whereas compounds like 190, 220, and 245 led to overall reduced labeling of many proteins. And we also tested this protein reactivity in the lyses of the primary T cells, including CD8 positive and CD4 positive T cells. We also evaluated the effect of the compounds on the cell viability as well. Some compounds seem to be somewhat more cytotoxic than the others, regardless of the cell type that we use at 100 micromolar concentrations. For compound 245 here, for some reason, T cells showed a higher sensitivity than other cell types towards this compound. We then performed the functional screening in primary human T cells, first looking for a compound that can inhibit the T cell activations measured by the surface expression of CD25 and CD69, and also the cytokine production. There are multiple hit compounds that show the inhibition of the CD25 and CD69 expression more than 60% at the concentration of the 50 micromolar, and also dose-dependent manner. Some of these active compounds also inhibited the T cell proliferation measured by the CFSE staining. When tested in the memory CD4 positive T cells, some compounds also showed the decrease in the cytokines, such as GM-CSF and the IL-2. One of the hit compounds, this compound BDHI-151, when we look at the structure of this compound, we noticed that actually there are 2 electrophilic centers. One obviously being that BDHI, and the other one, this 4-chloropyrazolopyridine moiety. In fact, when we incubated the compound with the glutathione or the cysteine, what we found was the covalent adduct formation by this chlorodisplacement mechanism. And when we removed that chlorofunctional group from the molecule, we didn't observe any covalent adduct formation after overnight incubation. With this in mind, as a kind of pilot proteomic experiment, we simply treated Jurkat with the compound 151 and searched for the protein modifications both by bromo displacement and chloro displacement through not only the cysteine but also serine and lysine. This was done without enrichment and additional labeling, thereby biased by the protein abundance, fragmentation and ionization. Nonetheless, we observed the covalent modification of a few proteins. So here, found in all 3 samples of the triplicates in bold, and the bromo displacement in yellow, and the chloro displacement in green. We saw some serine and lysine modifications. And one cysteine modification we found was actually at the cysteine 358 of the pyruvate kinase. This is particularly interesting to us because cysteine 358 is one of the major sites for the post-translation modification of the pyruvate kinase M. Pyruvate kinase is a terminal glycolytic pathway enzyme that catalyzes the transphosphorylation reaction between the phosphoenol pyruvate and ADP to yield the pyruvate. The high concentrations of the reactive oxygen species can induce an oxidation of this cysteine 385, which could result in the dissociation of the PKM2 to the less active dimeric form. And this can lead to an accumulation of the glycolytic intermediates. Actually, there are compounds reported to modify cysteines on the pyruvate kinase M, but not the cysteine 358. And depending on the modification site and the mechanism of action, compound could activate the PKM enzymatic activity or inhibit the activity. When we tested for the competitive labeling of the PKM2 using the IA camera again here, we found that compound 151 could partially block the IA probe labeling. And the IA conformation of the compound seemed to be more potent than the S conformation. And the deschloro analog that we had also showed some inhibition of the labeling, but at lesser extent than the parent compound 151. So we are currently validating to see whether compound 151 can actually modify the cysteine 358 of the PKM and what kind of functional effect that could have. But to more systematically map the protein targets and the sites of the modification, mainly focusing on the cysteine modification, we started using the competitive isotope activity-based protein profiling. In brief, cell lyses are treated with the either compound or the control vehicle in a DMSO, and the samples are then labeled with this reactive probe iorasamide alkyne, followed by the subsequent capturing using the click chemistry. After mixing control and compound 3D samples, probe labeled proteins are enriched, and then subsequent digestion followed by the analysis of the peptides by LC-MS could define the cysteine residues that show the changes in the MS chromatographic peak area comparing the compound treated and control samples, and that can define the liganded cysteines. So for our study, we used this isotopically differentiated desdiobiotin azide tech recently developed by Stefan Hecker group. And using this approach, we are able to identify the active cysteines in the GIRCAT proteomes. So this is data. So we identified about 440 peptides that compound 151 engages in binding. In this experiment, we set a slightly higher threshold for the ligand cysteines because the treatment with the compound was done at pretty high concentration of 500 micromolar for a long incubation time. Compared to a more reactive chlorasamide containing scout fragment, we observed that less of the peptide modification as expected, but somewhat selective binding of a compound to certain proteins such as cysteine proteases and E3 ubiquitin protein ligases. And one of the target proteins we identified is this DESI1, which I'll come back to in a minute. So from the competitive analysis between the chlorasamide scout fragment and our BGHI 151 compound, actually a lot of proteins are liganded by both compounds, as you can see here. And there is a number of proteins that showed a preferential binding to the compound 151, as you can see here. And since we are aware that our compound 151 can modify cysteines through two different mechanisms, we are currently evaluating 151 analogs, including this chloro compound, and also some other BDHI containing candidates that we identified in the functional screening, as well as lower concentrations of the 151 compound in the proteomic studies. Meanwhile, we started validating protein targeting of these BDHI compounds with the proteins we identified in early proteomics data as a potential target. One of those proteins is desmoylating isopeptidase 1, DC1, which belongs to a second class of the desmoylating enzyme, in addition to small specific protease families, which its cysteine protease activity, isopeptidase activity. It can deconjugate the ubiquitin-like modifier SUMO123 from some substrate proteins. And actually, DC1 is capable of desmoylating the transcriptional repressor B cell and is implicated in the T cell differentiation, activation, and also proliferation. But since its identification in 2012, DC1 hasn't been really studied much. The ligand cysteine that we identified with our compound 151 is actually the active cysteine residue that comprises the catalytic diet with the histidine residue in the active site. When we covalently docked our compound to X-ray crystal structure of the DC1, it appeared that our compound is well aligned in the binding pocket of the DC1, and we can actually see that the backbone of the cysteine 108, as well as the neighboring side chain of the asparagine 109, could participate in the binding and the stabilization of the dihydroisoxazole ring of the compound in the binding pocket. By competitive labeling of the cysteine, again with the IA camera, we found that the compound is able to block the cysteine labeling of the IA camera, and also the DES-chloro analog also is able to do so. Between the R and S conformations, again, the R conformation of the compound seems to be more potent. And this competition is well reflected of the information that we can already find in the literature using this, again, the reactive chloroacetamide scalp fragments. So we are currently validating and optimizing the biochemical assay for the DC1 to screen our BDHI compound library that we have, and optimize the potency and selectivity of the compound, because we know that our initial hit compound 151 can target multiple proteins in the proteins. Additionally, because compound 151 showed some interesting activity in the T cells and has got the two electrophilic centers, we've devised this alkylated analog to study mechanism of this compound for interactions with the proteins. So we first confirmed the alkyne analog is able to inhibit the T cell activation and proliferation, and our compound also shows the similar pattern of the protein binding compared to the protein compared to the parent compound 151, and also we confirmed the sufficient labeling of the proteins by using this alkylated analogs. So we are currently planning to apply this compound directly to the human cells to do more protein profiling study and understand the mechanism of action. Hopefully, we will be able to identify the site and the mechanism of the covalent modification and understand the biology behind this active compound. So obviously, there are a lot of things that we need to do. Definitely, we are trying to do some of the mechanistic studies of the active compounds that we found in that T cell functional screening. And we also like to assess the ligandability of many immune-relevant proteins that we identified in the initial proteomic study as a potential target. And also other plan we have is to kind of survey the protein targeting by this BDHI warhead more comprehensively to see whether it is true that it requires some local environment factors that could determine the covalent modification. And also, we really would like to expand the utility of these compounds by tuning the proteome reactivity and selectivity, and use the information that we gather from the proteomics studies that we are currently doing. With that, I'd like to also thank my people in the group. As I mentioned, Ray and David and Robbie really put a lot of effort and energy evaluating these compounds. And I also need to thank collaborators, the proteomics core, Ronald and Dorkel, for their support and the analysis of the LCMS data. And my colleagues here at the Chemical Biology Laboratory, especially Jordan Meyer and Jay Snicolet, for their support and help. Thank you for your attention, and I'm happy to answer any questions that you may have.
Thank you for this great talk. I see there are some questions from Stefan. Very interesting talk. Have you also tried the competitive gel-based ABPP assays with a probe based on your warhead instead of the iodine alkyne probe?
Oh yeah, using directly the probe that we have. So yeah, so the probe itself has very specific kind of binding preferences. So we cannot really use as a global profiling method to do the competition. But yeah, we've used that probe to do kind of assess the binding of some of the heat compounds that we identified, mainly using the recombinant enzymes, but not in the lysate or the intact cell base.
And a question from Stefan Sieber. Did you compare the influence of the halogen leaving group of the warhead on the proteome reactivity?
Yeah, we definitely are thinking of doing that. Definitely we want to tune the reactivity by changing the halogen group there, bromo to chloro, and also potentially putting some other electronic functional groups on the ring system itself.
Okay. I was wondering if you compare your warhead to the more standard warheads like the acrylamide or butinamide, how would they compare? They are less reactive, I assume?
Yeah, definitely less reactive. But, again, the acrylamide has very mild reactivity. So I think the chemical reactivity could be somewhat similar. We don't see a lot of, chemical or solution reactivity of the warhead itself. But that's why we are thinking that it may have a very specific protein, reactivity. But yeah, at least for the chemical reactivity, they are somewhat similar to the very mild acrylamide warheads.
And have you ever used this warhead in an in vivo situation? Is it stable, for example? Does it get oxidized in the liver? Or?
We haven't tested that yet. With the alkyne analog that we have, we are certainly planning to apply directly to the in vivo or other more complex cell systems. So we'll hopefully know the stability in the environment.
Okay. Are there any further questions? If not, then I thank you again for this wonderful talk. And then we will move on to our next speaker. And the second speaker of this session is Dr. Chris Parker. If you could share your screen, please. He's an assistant professor at the Department of Chemistry of the Scripps Research Institute, and he received his PhD in chemistry and chemical biology from Yale University in 2013, and was a postdoctoral fellow in the group of Professor Ben Cravat. And today he will talk about a new technology to study the effect of acetylation on specific proteins. So please, Chris, the screen is yours.
Thank you. Can you hear me and see my screen? Okay.
Yes.
Great. Well, thank you, Mario, for the introduction, and thank you to Stefan Hacker and Sieber for the invitation, as well as to the organizers for hosting the symposium. And a big congrats for them as well, considering this is an inaugural event. It's been really great listening to everyone's science, and I'm looking forward to watching the remainder of the talks. By way of background, I started my lab almost exactly 3 years ago, at least last month. And broadly speaking, we're interested in developing useful chemical probes to study protein function in the context of human disease. And to enable us to do that, we rely quite heavily on the use of mass spec-based proteomics. I'd say there's kind of 3 tiers of research in our lab. One, the first is sort of the bread and butter of our lab, which is chemoproteomic inhibitor discovery, methodology development, and so on. We do quite a bit of target ID of bioactive compounds, such as metabolites and screening hits. In terms of targets, we're interested in using our methods to pharmacologically access protein families that have presented challenges using traditional strategies. We're also interested in using our platforms to create druggability maps of cell states. That is, to identify targets that might be uniquely accessible to small molecules under specific contexts, with the overall idea of being able to therapeutically target proteins in specific contexts. And lastly, we're leveraging our ligand discovery technologies to develop bifunctional compounds to selectively modulate PTMs on targeted proteins. And like many labs, we're of course interested in targeted ubiquitination, but we're also looking beyond to other PTMs. And I thought towards that end today, I thought I would move a little bit away from chemical proteomics for a bit and actually focus on a newer area of our lab, which was mentioned, the development of bifunctional molecules that are capable of modulating protein acetylation selectively on targeted proteins. And so this work is very much a work in progress, but I'm looking forward to sharing where we're at. First, just a little bit of a background. If we look at some of the most frequent PTMs, phosphorylation, ubiquitination, acetylation, and N-link glycosylation, we see that PTMs on thousands of sites are often governed by a surprising number of writers and erasers. Writers and erasers meaning the enzymes that put the PTMs on and take them off. For example, there's experimental evidence for around 230,000 phosphosites occurring on anywhere between 13 and 15,000 proteins. This number's certainly growing, but this means roughly that for each kinase, there's 450 phosphosites, and for every phosphosite or phosphatase, there's around 1,000 phosphosites. This is, of course, an oversimplification of these numbers, but it still provides a general kind of ballpark overview. And the same is true for other high-frequency PTMs, particularly for acetylation, where there's only around 23 acetyltransferases and around 18, 19, or 20 deacetylases. But currently, there's around 20,000 experimentally measured sites of acetylation so far on around 7,000 proteins. So there's about 1,000-fold difference there. And so overall, this implies that PTM enzymes are promiscuous and they control multiple signaling pathways. And so when we think about ways that we might want to control these pathways by regulating these enzymes, like with inhibitors, we're not only affecting the pathway that we care about, but we might be affecting hundreds, if not thousands of others. And so like many other labs, these numbers got us thinking of ways in which we can go about selectively modulating PTMs in a way that's useful to study their function. Our lab is particularly interested in acetylation. And so as a brief refresher, lysine acetylation occurs through the lysine acetyltransferase, or CATS, catalyzed transfer of acetyl group from acetyl-CoA to the epsilon amino acid side chain of lysine. Alternatively, acetyl-CoA can acetylate lysine in non-enzymatic fashion, but I won't really talk about those today. Acetylation can be removed by histone deacetylases, which are zinc-dependent, or NAD-dependent sirtuins, which are deacetylases. Lysine acetylation was discovered more than half a century ago as a PTM of histones and has been extensively studied in the context of transcriptional regulation in that regard. Here, acetylation of histone tails induces a charge change, which can disrupt nucleosome formation and make DNA more accessible for transcription. But over the past decade, advances in mass spec-based proteomics have expanded our inventory of acetylated proteins to include thousands of non-histone proteins that regulate key aspects of most cellular processes, like, of course, transcription, but metabolism, signal transduction, as well as protein folding. Acetylation can do this through diverse mechanisms, which include modulating enzymatic activity, so they could enhance or inhibit enzymatic activity, or even modulate substrate specificity. For example, acetylation inactivates glucose 6 phosphate dehydrogenase by blocking dimerization. Acetylation of MDM2 recruits the deubiquitinase USB7, which deubiquitinates MDM2 and redirects its activity towards another substrate, P53, resulting in its degradation. Acetylation can also modulate other lysine modification. It can also communicate with other lysine modifications, as I mentioned, ubiquitination, but also methylation and others. It can also modulate various protein-protein interactions, as well as affect protein localization. So there's this really neat study that was just published a few months ago where acetylation of PDL1 blocks nuclear translocation and transcriptional activation. So what makes us really excited about acetylation, it often represents a turn-on signal for protein, or maybe even a change of function for the protein as opposed to direct inhibition. So this, to us, represents a whole new way to modulate proteins and small molecules. So if we take a look at the classical way we study and modulate protein acetylation, as well as many other PTMs, it's often through the blockade of writers and erasers, as I kind of discussed earlier, which can have non-specific effects, global effects on all substrates. Of course, mutating substrate sites to either block or mimic acetylation, but this is irreversible typically, and it lacks dynamic and temporal control. Lastly, there's a variety of ways to study acetylation biochemically and in vitro through the synthesis of substrates or the use of non-natural amino acids. But very often, these studies aren't very amenable to cell-based systems and again, generally lack dynamic control. So encouraged by the transformative utility of other bifunctional molecule systems, such as chemical inducers of dimerizations, ubiquitin-inducing small molecules, LITACs, as we'll hear about later on, as well as recent examples of bifunctional small molecules that can modulate phosphorylation, we envision a similar heterobifunctional system could be developed to modulate acetylation on any protein of interest on demand via chemically induced proximity. And we're particularly interested in inducing acetylation, so adding an acetyl group to proteins, and therefore we would need to find molecules that can bind and recruit acetyl transferases to a protein of interest. Among the CATs, the E1A associated protein P300 and its paralog Krebs binding protein, or CBP, are kind of the most prominent acetyl transferases in humans. They collectively regulate greater than 2/3 of all known acetylation sites in humans. And so to us, this suggested potentially broad substrate tolerability, and therefore represent a high priority candidate for the design of these bifunctional molecules. Fortunately, various potent and selective ligands targeting the bromo domain, which is conserved among P300 and CBP, have been developed, and they don't appear to greatly affect the catalytic acetyl transferase activity. And particularly, a series of benzimidazole based inhibitors developed by Paul Brennan's lab show quite selective potency, quite good selectivity and high potency in the low nanomolar range against P300 CBP. And structural studies suggest that we could append a linker to this methoxy group to our target binding terminus. In addition, I should mention that recent studies from Nathaniel Hathaway and others have also co-opted these inhibitors into bifunctional molecules to target DNA either through polyamides, so targeting DNA directly, or indirectly through the use of inactivated Cas9 to modulate gene transcription, so to recruit P300 to transcription sites. But we thought we could actually adapt these molecules and derivatize them with linker to direct P300 CBP to target proteins. Thinking about the target binding ligand, a major challenge here is that acetylation is often an activating modification. It certainly could be inhibitory. However, most pharmacopoeia is usually directed towards inhibition, so we had these counteracting activities. To overcome this obstacle, we decided to instead adopt a protein tagging strategy. Specifically, we've used target proteins with an F36V mutant form of FKBP12, which is a small tag and has small potent selective small molecules available, which have also been recently co-opted by Jay Bradner and co-workers to direct E3 machinery to target proteins, resulting in degradation. This is called DTAG. This would allow us, in theory, to target any protein for acetylation in the absence of available ligands. And so we appended a previously reported FKB12 binding ligand to the bromo domain inhibitor that I mentioned before. We developed a number of different linkers. I'm just showing a handful here. The first thing we want to do is actually confirm that these bifunctional molecules can engage the targets in cells. So for that, we made photoaffinity probes of both the FKB binding site and the P300 binding site and did competitive profiling and showing indeed these ACE tag molecules can bind to the targets we want them to bind to in cells. We next sought the ability for these ACE tag molecules to mediate ternary complex between soluble FKBP12 and the bromodomain part of P300 in vitro using AlphaScreen. We were quite excited to see a characteristic bell-shaped auto-inhibitory curve for all ACE tag analogs. And again, this is the result of ACE tag molecules effectively outcompeting ternary complex formation. Although we didn't explore too many linkers, we know that there's different linkers promote varying levels of ternary complex, and we chose PEG2 linker based off of this data, as well as some cell-based data. We next confirmed that the ternary complex can be outcompeted by each of the ternary binding ligands, showing that binding to both sites are important or necessary for protein or for ternary complex formation. We next evaluated whether ACE tag molecules could actually induce acetylation of tagged proteins in cells. And so we first chose to target histone H3.3, given that P300-CBP is a predominant histone acetyltransferase. And so using HeLa cells, we stably transfected the H3.3 FKBP12 chimera, and we were quite delighted to see that ACE tag molecules can induce a nice dose-dependent acetylation of H3. Specifically, we see induced acetylation at lysine 18, 23, and 27, which were reported to be the predominant targets of P300-CBP through other studies. Although it's not as obvious in vitro as it is in vitro, we see a predicted hook effect at higher concentrations of ACE tag. Notably, acetylation is maxed out at submicromolar concentrations, so around 500 nanomolar. Further, this acetylation can be blocked with competing ligands. So if we add competing ligands both on the FKBP side and P300 sides, the acetylation goes away. We could also add catalytic inhibitors of the acetyltransferase and it's blocked. I'll elaborate on this a little bit later, but we also observed no effects on endogenous on-tagged histones. We next wanted to assess the kinetics of induced acetylation. So we performed a time course. And unexpectedly, so this is set up 5, 10, 30, 60 minutes, plus minus the ACE tag molecule. Unexpectedly, we observed target acetylation occur quite rapidly within 5 minutes, which is about as fast as we can actually go to treat the cells and then quench the activity. So presumably it's happening faster than that. And acetylation seems to continue beyond that time point. And the longest we've ever looked at is 24 hours, and it's still going strong. This is in the presence of the molecule. Notably, when we perform washout experiments, so we treat the cells and then we put in fresh media without the molecule in there, we see that acetylation slowly decreases down to basal levels in about 2 hours. And we think this is likely the result of endogenous lysine deacetylase activity, showing that it's dynamic and a reversible modification. Next, we wanted to assess the selectivity of induced acetylation. For this, we turned to mass spec-based proteomics. Given that selective acetylysine antibodies aren't really available for all sites on H3, we first wanted to assess the selectivity on the targeted protein in an unbiased fashion. So in these experiments, we treated cells, we enriched the target by the tag, which is pretty convenient, and then we monitored acetylation of the tag protein by quantitative mass spec. And in these experiments, we showed that acetylation of K18, 23, and 27 is quite dominant. So this is shown with the blue dots. And we don't detect any acetylation at other sites. We use SAHA, an HDAC inhibitor, as a positive control, just to show that even though ACE tag doesn't promote acetylation at these sites, SAHA does. And this was consistent with our Western blot studies. And the cell, which is a nice workflow to monitor acetylation independent of antibodies. We next wanted to look proteome-wide. So here we use a polyacetylysine antibody to enrich acetylated peptides. So this is just sort of a standard workflow for acetyl proteomics. We monitored acetylation levels over thousands of proteins, and we found that apparently no other protein had substantial changes in acetylation. So this is shown in this top volcano plot. I should mention that although we detect endogenous H3 in these cells, we don't actually see the FKBP tag version by mass spec. And I'm not really showing it here, but the endogenous, as one might expect, the endogenous levels of H3 are 25, 30-fold higher expression than our tag version, which is not resolvable by mass spec. So this is why we have to do the enrichment on the target. We also monitor acetylation of cells treated with SAHA and see dramatic changes shown here in red. So together, this data suggests that our ACE tag method is highly selective for FKBP tag proteins. Satisfied with the successful compound-induced acetylation of H3.3, we next used our system to assess the acetylation of other targeted proteins, specifically P65 and P53. P65, or REL-A, is a subunit of NF-kappa B, and acetylation is at various sites has been shown to regulate the stability and transcriptional activation of P65, although the details of that are not really firmly established. So we first generated, once again, a stable expressing FKBP12 REL-A chimera cell line and P65 knockout HeLa cells. And we show that ACE-TAG, once again, induces a strong dose-dependent acetylation of P65, but mainly at lysine 310, which is a site previously reported to induce transcriptional activation. And although I'm not showing it here, we do have some preliminary data showing that this might affect NF-kappa B activation in simple luciferase reporter assays. And I'm also not showing it here, but the kinetics of both P65 and P53 are very similar. So we see acetylation induced within a matter of minutes. Acetylation of P53 is extensively reported to induce transcriptional activation and subsequently activate the metabolic, self-regulatory, cell cycle, and apoptotic functions of P53. P53 is actually the first non-histone substrate of P300 CBP identified, and it's now reported that 4 or 5 other acetyltransferases might have some roles. There are about 13 lysine residues thought to be modified by acetylation on P53, and the majority of them are kind of clustering around the C terminus here. And as I alluded to before, there are many publications in this area. However, the functional consequences of acetylation of these sites aren't really well defined. Some reports suggest that acetylation of all 8 terminal lysine residues are required for activation, while others suggest it might be 1 to 2. Some sites are reported to result in protein stabilization and recruitment of co-activators and so on. Often these studies are conducted by inhibiting or overexpressing the CATs, which could have global effects or through mutations, or which could obviously affect through other PTMs or result in compensation, especially when you have these clustered sites. If you mutate one, the enzyme might hop over to another one, so they might be redundant. So using our method, we made both the C and N terminal tagged versions of P53 in NSCLC cell line H1299s, which are naturally null for P53. And once again, we show that just like with other targets, we see a strong dose-dependent acetylation at 3 different sites, 305, 373, and 382. Interestingly, we observed the same selectivity on both the N and C terminus of tag P53, suggesting that these might actually be predominant substrates of P300 CBP, at least in the system. And once again, we see sort of this hallmark bell-shaped curve. Given the strong induction, we wanted to explore whether if there were any resulting functional consequences. Considering the diverse gene products previously reported to be controlled by P53, we took a look at 4 of the predominant P53 target genes, kind of representative of the 4 general functional roles of P53. And this includes P21, which is a CDK inhibitor and regulator cell cycle, PUMA, which is a pro-apoptotic BCL2 protein, GLS2, which is a metabolic glutaminase, and MDM2, which is the negative regulator of P53. Shockingly, we see quite selective, at least among these 4 genes, induction of just P21 by itself, both by protein levels, but also transcript levels. And notably, these effects weren't observed in control cells, and we show P21 expression is dependent on catalytic activity. It can be outcompeted. And we interestingly observed the same effect in both the N-terminal and C-terminal cells, tagged P53 cells, suggesting that this, again, might be the predominant substrate of those might be the predominant sites of P300. Working with Michael Erb's lab, we began to look at this a little bit more globally. So we performed RNA-seq on ACE-TAG treated FKB P53 1299 cells, as well as wild type cells, and see surprisingly clean transcriptional changes. Although gene set enrichment analysis revealed on-pathway enrichment of P53 genes, we see only very minor increases in target genes that affect apoptotic or metabolic or regulatory functions of P53. Taking a look at the data a little bit differently in these volcano plots, we see actually a very clean enrichment of P21 or CDKN1A transcripts, as well as this other protein called GDF15. These are about 3-fold enriched in treated cells versus DMSO treated cells, and we see no effect in control cells. We also confirm that this protein GDF15 is increased both on the protein level as well as the transcript level. Not much is really known about GDF15. It's recently annotated as being a p53 target gene. It's secreted and appears to affect cell motility, but interestingly, there's not really any reports suggesting that its expression is regulated by this acetyltransferase. So P21, on the other hand, is a principal target of p53 transcriptional activation, directly binds and inhibits CDKs, which are required for cell cycle regulation. Upon exposure to various perturbagens, such as DNA damaging reagents or oxidative stress, it's thought that p53 is acylated, activated, leading to p21 expression and arresting cell cycle. However, these stressors, which are often UV irradiation or DNA intercalators, are often nonspecific, and this can lead to variable p53 acetylation and activation of other downstream p53 targets, resulting in apoptosis as well. And so the impact of acetylation on these diverse activities of p53 aren't really well fleshed out. So we thought we might use our system to study it a little bit more. We first showed that AceTag1 had no significant effects on cell viability or on apoptosis, looking both at caspase-3 and annexin staining. And this is consistent with our RNA-seq data showing that we don't really affect any apoptotic genes when we induce acetylation of p53 with AceTag1. Given that AceTag1 induces p21 expression, we next investigated whether effects on cell cycle were to be had, and we found that treatment of the few chimeric 1299 cells with AceTag1 led to substantial amplification, 10 to 20% of cells in the G1S phase. Importantly, these effects were not observed in control cells. We recognize this effect is not dramatic, but we think that's probably likely more the result of the relatively low expression levels in our system. So these are stably overexpressing cells, so they went through a selection process. Nonetheless, our data suggests that chemically induced proximity of p53 and p300 CBP results in p53 acetylation, primarily p21 and GDF15 induction and subsequent cell cycle arrest. However, it doesn't seem to modulate the apoptotic functions of p53. And so it's tempting to speculate that, at least in this system, this might be the predominant role of p300 CBP in regulating p53 activity, which might be lost in other studies using less selective modulators of acetylation. So this data is unfolding as we speak, and hopefully we'll have more to come. So in summary, we've been able to show that AceTag molecules can controllably induce acetylation on targeted proteins in live cells. It's rapid. It occurs often within minutes upon molecule addition. It's selected for tagged proteins. It's reversible and dependent on enzymatic activity, and it possesses submicromolar activity as well as all the kind of characteristics associated with heterofunctional biomolecules. We think AceTag will be useful tool to study the dynamics of acetylation on targeted proteins. And as you kind of saw in our last story, we're really interested in p53. But we're also using our system to investigate the roles of other proteins such as BCL6 and FOXP3 and PDL1. So proteins that are heavily implicated in cancer and immune disease. We actually think this could be a useful tool to screen targets to identify potential targets that might have some sort of therapeutic value through modulation of acetylation. And we envision that if induced acetylation using AceTag results in disease combating functional consequences, we could use existing small molecules or our ligand discovery platforms to develop ligands that bind to endogenous forms of these proteins, so-called AceTags. With that, I'd like to thank my group, collaborators, and funding sources. Specifically, this work is a product of the group members in bold, Wesley, Pazo, Jacob, and Annie, really spearheaded by Annie, who's a graduate student at Wesley, a postdoc in the lab. And I also like to thank Herb Lab for the help with the transcriptomics, and I'd be happy to answer any questions.
Thank you, Chris, for this exciting presentation. There are several questions, so I start with Stefan. Stefan Hecker, as you use tags on the protein of interest and at the protein modifier, would you assume that this approach can be modularly applied to other PTMs installing or removing enzymes?
Yeah, absolutely. So I think this could be a more generalizable approach, and people have done it, I believe, with phosphatases as well. And we're pursuing deacylases, as I know other groups are. The big challenge with recruiting PTM machinery is just finding ligands that actually don't affect the PTM editor function at all, which most ligands that we have are inhibitors. So it could be a challenge there.
Okay. From Timo Wendel, thanks for the interesting talk. Do you think the observed acetylation pattern of the AceTag-induced acetylation is due to the intrinsic selectivity of the proteins or rather directed by the AceTag?
Yeah, I think that's a really great question, and it's something that we're aggressively kind of seeking the answer to. One trick that we kind of play with all the targets is we put the tag on both the C and N terminus, and it seems like the selectivity is maintained kind of independent of where we put the tag, at least at those two positions, which might suggest that substrate selectivity is dictated by the enzyme itself, that maybe we can't force it to acetylate certain sites. But this was N of 3. So this is something that we're pursuing, looking at different linker structures, trying to using ACE tags, recruiting the acetyltransferase to different parts of the protein. But right now, all of our data suggests that it's a kind of a state function of the enzyme as opposed to being able to site selectively modulate acetylation.
And as a follow-up, if you block the HDAC, for example, would you then see, for example, increased acetylation than if the HDACs are actually rate limiting the activity?
Yeah, that's interesting. So if you block HDACs, at least the targets that we looked at, which are also substrates for those deacetylases which HDAC inhibitors hit, you just see massive amplification of acetylation. So it's hard to find this kind of gravy area where you're inhibiting HDACs enough where you're not removing, within the same time frame the induced acetylation without maxing out sort of the stoichiometry of acetylation. So it's a tough window to find, but if you add both HDACs and ACE-TAG, you definitely get even greater combined amplification of stoichiometry. But at that point, it's just really hard to tell, just because when you inhibit HDACs, you just see massive amounts of acetylation.
Okay, yeah. There are some more questions from an anonymous attendee. As you tested various linker lengths for your ACE-TAG approach, did you find some guidelines for optimal or minimal linker length needed to achieve successful target acetylation?
Yeah, so it seemed that each target had its own subtle linker preferences. We've only explored a handful of linkers at this point, which I think this is the next step if we want to get serious about these sort of bifunctionals, that we're going to have to really delve into the linkerology a bit. But it's dependent both on linker length and composition, which is perhaps not surprising, as those lessons have been learned with other bifunctional molecules.
Okay, and then Laitang Liang has a question. Are there any proteins that are currently suspected but not confirmed to be acetylated? Yeah,
I mean, there's computational tools that predict based on substrate knowledge, sequence knowledge, to be predicted. But this is an interesting question to target proteins that are not known to be substrates of any acetyltransferases, but might have acetylatable lysines. This still needs to be done. This is a new area, so I think that's a great question and exciting area to think about.
Okay, and then a question from Stefan Sieber. Exciting story. Thank you, Chris. Could you please explain if the fusion between the FKBP and the protein of interest could affect the acetylation efficacy? Is there a limit of the protein of interest size or the localization of the respective lysine residue?
Yeah, it's an interesting question. So we studied a little bit further on H3 to see if it affected localization and things like that, and we see it actually still gets incorporated in chromatin. But anytime, of course, you put a tag on a protein, you could be affecting structure, function, localization, and things like that. So it's a major drawback to this approach, or any tagging approach. But I didn't catch the second part of that question.
Whether the localization of the lysine would also be a limit or an important factor.
Yeah, I kind of go back to the C and N terminal experiments where we put the tag on both the C and N terminus of all three targets. And again, we saw the very consensus among the sites that are selectively being targeted. But again, this is three targets that we've explored so far. But they seem to be, these are all known substrates of this enzyme. So it could be in this instance, as we're not using neo-substrates, that maybe we can't affect the substrate selectivity very much. But maybe with other systems or non-native substrates that we might be able to tune the selectivity a little bit more.
Okay, then I have one final question. You said that your compounds induce cell cycle arrest, but did not induce apoptosis. Do you think these type of compounds could induce cell senescence?
Yeah, exactly. So that's exactly what we're looking into, and we're looking into frequent p53 mutations that are deactivating to see if we can potentially rescue p53 activity. But yeah, in terms of phenotypic characterization, we've mostly only looked at, we're honestly in the beginning of this study, we're just trying to confirm all the literature around p53 acetylation. We're expecting apoptosis, we're expecting sentience, we're expecting cell cycle rest and metabolic perturbation, but we were quite surprised when we saw this very narrow window of activation. And that, again, that's sort of a recent finding that we've only had a little bit of time to dig into. But it's, you know, it's not dramatic. So I think the right experiments, rather than using these stable expressing cell lines, would be to knock in the tag to wild type p53 in cells, that cancer cells that actually have wild type p53, rather than these null cells. But active area of investigation for sure right now.
Okay, so thank you again for sharing your exciting results with us. And then I think we have a 15-minute break, and then we resume with the last session of today. So I will see you in 50 minutes. Thank you.
Great, thank you.
Yeah, so welcome everyone to our final session today. It's a great pleasure. My name is Stefan Sieber from TU Munich, and I'm welcoming first our next speaker, which is Carrie Bacchus, who is giving the first talk, and I would like to briefly introduce Carrie. So she is an assistant professor at UCLA, and she studied at Brown University, did a PhD in the lab of Ben Davis and Cliff Berry, worked on mycobacterial trehalose-based chemical probes to label these microbes. And she did then a postdoctoral study at Ben Kravitz's lab at Scripps. And here she did groundbreaking research in cysteine and lysine labeling. And currently she's introducing a lot of exciting chemoproteomic tools. And today her talk is called, or is entitled, Expanding the Activity-Based Chemoproteomic Toolbox. So Carrie, we are looking very much forward to your presentation.
All right. Well, thank you, Stefan, for the wonderful introduction. And I wanted to say thank you to all the organizers, and particularly to Stefan Hacker for organizing this meeting and the invitation to talk. Let me just share my screen. All right. That look good?
Looks very good. Thank you.
All right. So it's really been an amazing early morning meeting. You know, it takes a lot to get me out of bed at 6 a.m. And I jumped out of bed to hear Ben Kravitz's talk and some of the really exciting work that's coming out of the Kravitz lab. And I was just delighted to see the work, particularly on NONO, because that was a protein that had intrigued me since working in Ben's lab of what that cysteine and that sort of beautiful pocket is doing. And so it's cool to see that come to light. So my lab is focused really on tool development and trying to make chemoproteomics better, faster, stronger. And we're excited about method development because of the fundamental druggability gap that I think a number of people in this meeting have already alluded to, and that's that, you know, there's roughly 20,000 odd protein coding genes. About a third of them have been linked to one or more human disorders, and really only about 3%, give or take, of the human proteome has been pharmacologically targeted by FDA-approved drugs. And so clearly there's 97% for this community to start thinking about new tools and technologies and, you know, new electrophiles and chemotypes that could access this remaining broad spectrum or swath of the undruggable proteome. And one feature that we find really exciting and exciting for a number of reasons, including some alluded to earlier, is developing molecules that target the thiol side chain of cysteine residues. And we like cysteines for several reasons. Most notably, nearly all proteins contain at least one cysteine. So it's a universal feature that really you could think of almost as an Achilles heel of tough to drug classes of proteins. And while, you know, if you think back to your introductory biochemistry class, probably the number one thing we learned about cysteines is that they are important for disulfide bonds and redox active and structural disulfides. A large fraction of cysteines are actually found in a reduced state. And this, you know, number is probably even higher when you look at intracellular and cytosolic proteins. So there's lots of reduced cysteines sort of waiting, lurking in the proteome, waiting to be drugged. And, you know, as Ben introduced earlier, there's beautiful examples of covalent cysteine reactive molecules that are actually approved for the treatment of a variety of indications, including several cancers. And, you know, the recent work coming out of Amgen and Wellspring showing that the undruggable protein KRAS could be targeted using these molecules that access an acquired cysteine, in this case, the glycine 12 cysteine mutated form of KRAS. So as a postdoc in the Kravitz lab, I set out to really try to answer the central question, I'd say, that, you know, eats away at a lot of us in this field is, you know, if this works for these beautiful compounds, it should work for more proteins. And if so, how many and which ones? And again, as Ben introduced, I developed a chemoproteomic platform that assayed the ligandability of cysteines proteome-wide and really did sort of a fishing expedition to try to find potential druggable sites. And what was really exciting from this study was that we identified not only a number of ligandable cysteine residues, so about 700 found on just over 600 proteins, but the overlap between the targets of these molecules and the targets of FDA-approved drugs was essentially negligible. So this sort of indicated to us that our library was accessing new druggable space and sort of closing this 97% druggability gap. And subsequent to that work, I'd say there's really been an explosion of cysteine reactive and more broadly covalent electrophilic small molecules in chemical biology, including electrophiles targeting different amino acid side chains, covalent PROTACs, as well as more recently covalent immunomodulators. Again, some beautiful work from Katja Vinogradova, as well as Megan Blewett coming out of the Cravat Lab and many others. And so when I started my group at UCLA now just over 3 years ago, I had sort of two big questions that were eating away at me and I wanted to really dive into. And one was really still getting back to this question of how many cysteines are druggable. You know, is it 700 as I showed you, or is it more than that? And you know, what is the limit of detection here and what's sort of slowing us down in terms of identifying reactive and ligandable cysteines? And the second, and I'd say this is a much bigger and much sort of less tractable question, is how do you pinpoint which of these many cysteines are functional? You know, how do you take the cysteine in no-no and know that that regulates the androgen receptor, for example? And, you know, are there ways that we could be smart about high throughput identification of functional cysteines such that it doesn't take, you know, multiple years to characterize each residue? And so for most of my talk, we'll talk about expanding the scope of the cysteinome, and then we'll dive into this question of functionality at the end. So when I started my group, one of my first graduate students, Maria, asked me what at the time seemed like a very simple but very elegant question. And so she said, you know, how many cysteines are there in the human proteome, not just in the residues, not just in the proteins that we detect, but in all proteins? And so she wrote some code and counted the cysteines, and it turns out there's a lot of them. So 260-odd thousand total cysteines. You know, this number was going to be slightly different depending on the particular proteome you consider. Of those, in my original chemoproteomic study, I'd identified about 2.5%, and about 0.25% were what we considered high confidence ligandable sites. And so you can see clearly there's a big gap here. And the question then becomes, why are we only sort of scraping the surface of the cysteinome, and how can we do better? And that's really what my lab, one of the big pushes in my lab has really been to sort of start to explore the rest of this portion of the pie chart. And so to think about how to make chemoproteomics better, you need to sort of familiarize yourself with the standard chemoproteomics workflow, which we've already seen several times today. Take cells or lysates, treat them with a compound, capture with a pan-cysteine reactive probe, do some bioorthogonal chemistry, and then either a gel-based profiling experiment or a proteomics experiment. And if you've ever done these experiments, you'll know that they're actually fairly tricky, and especially you know, for a young graduate student to set this up and get it working in a new lab can be a non-trivial exercise. And some of the issues for chemoproteomics stem from, you know, we say these cysteine reactive probes are pan-reactive. They're actually not. You know, a bioorthogonal chemistry. Kudos to Ben Cravat and Carolyn Bertozzi. To, you know, I have the fortunate opportunity here of introducing for developing this amazing chemistry. But it's not perfect. And, you know, so if you've dived into this proteomics data in any depth, you'll know that the reaction often does go to completion. And then there's challenges associated with recovery of biotinylated peptides. You know, biotin is a molecule with sort of challenging solubility profiles, and so sometimes just resolubilizing the peptides can be challenging. And so to get at some of these problems, we need to take a brief detour and talk about some new bioorthogonal chemistry that comes out of my lab. And this may seem like a bit of a detour, but I promise you it's really important for setting the stage of some of our more recent work. So Jian Cao, who is a postdoc in our group, asked the question whether or not we could swap out the alkyne handle for an aryl halogen and use a Suzuki-Miura bioorthogonal chemistry to conduct chemoproteomics experiments. And we have some exciting reasons for this that I don't have time to tell you about today, but suffice it to say that we wanted to develop this chemistry in the lab, and we had this vision for potential applications. And some of our motivations stem from the following. First, aryl halogens are really ubiquitous in drugs, particularly aryl chlorides and aryl fluorides, but also to a lesser extent, aryl iodides and aryl bromides. They're synthetically quite tractable. You know, at our favorite vendor down in San Diego, there's upwards of 30,000 aryl halogens that are commercially available compared to about 300 alkynes. And they're quite small. So, you know, a nice feature of a bioorthogonal handle is size should be minimized to reduce the chances for modulating or aberrantly impacting the function of the molecule. And aryl iodides are about the same size as alkynes. So with that motivation, we dove into the literature. And, you know, fortunately for me, I'd done my PhD in Ben Davis's lab when Justin Chalker and Chris Spicer and others were developing some of the early bioorthogonal Suzuki-Miara cross-coupling chemistry and showed that this chemistry works on cell surfaces as well as on recombinant proteins. There's also work showing this chemistry works on oligonucleotides, but no one had extended this chemistry to more complex systems such as whole cell lysates at the time of us starting this project. So Gian went and made two simple model probes, an alkyne and aryl iodide, cysteine reactive iodocetamide probes, and then set up quite quickly activity-based protein profiling gel-based assay. So here is shown a streptavidin blot comparing cross-coupling conditions to click chemistry conditions. And really in a matter of weeks, we'd identified catalyst conditions that we could get really high cross-coupling, low background, and it all looked really promising. And so then, you know, we were excited to jump on the mass spec and compare these two probes, and spent a number of months struggling with this portion of the project. What seemed like it should be quite simple, would label lysates using the Suzuki workflow that worked beautifully for the streptavidin blot, and Gian would get no labeled peptides or proteins in his mass spec samples. And then he would do the control experiment and get, you know, upwards of 5,000 odd cysteines in a single shot proteomics experiment. So we knew the mass spec was working, but something was going horribly wrong with the Suzuki chemistry, and we scratched our heads, and we're lucky to be down the halls from another proteomics lab at UCLA, and they suggested that potentially we should try out this new solid phase extraction method called SP3 sample cleanup. The way this method works is it uses these carboxyl-coated magnetic beads to enrich proteins and peptides from solutions contaminated with all sorts of common biomolecules. And lo and behold, we tried this method, and our hypothesis was that our samples were contaminated with excess of the biotin boronic acid, and if we could deplete that, that would improve our coverage. And lo and behold, that is indeed what happened. So we not only increased our coverage of cysteine-containing peptides identified by click chemistry, we also dramatically increased the coverage of peptides identified by the Suzuki-Miara cross-coupling chemistry. So you can see we went from essentially 0 peptides to upwards of 7,000 unique peptides in a single-shot mass spec experiment. And so this was really exciting to us because it pointed to not only a way to develop new bioorthogonal chemistry, but also a more general approach to improve our cysteine chemoproteomics. And so I should say this work was recently published in Analytical Chemistry if you're interested in more of the details. What a graduate student in my lab, Sunny Yan, did was then return to this question of how do we make cysteine chemoproteomics as high coverage as we possibly can. And I alluded to this earlier, but we discovered that the combination of our standard labeling conditions, so 100 micromolar of iodocetamide alkyne and 100 micromolar of biotin azide, only achieved about 30 to 40% biotinylation of labeled peptides. And Sunny optimized these conditions by increasing the labeling reagents as well as some other adjustments, and could push this chemistry to closer to 100% efficiency. And so that together with our new Orbitrap Eclipse tribrid mass spectrometer, which is equipped with this FAMES ion mobility device, allowed us to increase the coverage of PSMs from about 10,000 in a single shot 70-minute gradient to upwards of, I'm not sure why this is not showing, but about 25,000 peptides, cysteine-containing peptides in a single experiment. And so with that, we then asked the question, how many cysteines could we identify? And returning to that original number, we'd identified about 2.5% in my study from 2016. In Sunny's study, she achieved 13% of all cysteines in the human proteome. And then we also counted the total number of cysteines that are theoretically detectable, meaning that are in tryptic peptides of an appropriate length for detection using trypsin as your sequence-specific protease. And so clearly there's still a long ways to go to get to full coverage of the human proteome, but we've achieved about a 5-fold increase in coverage. And what was really exciting to us as we compared our work to a recent study by Steve Gigi's lab, not only was our study, accessing slightly more cysteines in a less instrument time, the overlap between our study and the Gigi lab study was actually relatively modest. And so this probably stems in part from different cell lines that were used, as well as different choice of reagents. But really what this goes to show is that fairly simple changes to your proteomics workflow can actually dramatically increase your coverage of detectable cysteines. And so more recently, my lab has taken this information and pushed forward to really start to dive into the proteomics in a little bit more depth. And so this was a team effort, again, by Sunny and a first-year student in the lab, Andrew. And we asked the question whether we could learn anything about the fragmentation of these labeled peptides and apply that to further improve our coverage. And so, if you know anything about tandem mass spectrometry, we typically take cells or lysates, proteolyze the proteomes, and subject them to biotinylation conditions. In the gas phase, we first collect a full mass spec scan, so an MS1, detecting the mass to charge ratio for a labeled peptide. Then these peptides are fragmented, and we detect the B and Y ions for pieces of the peptide, and this information is then put back together to figure out the likely sequence of the peptide of interest. And so our question was, are there any ions in these spectra that potentially aren't identified by a standard data analysis workflows that maybe could be leveraged to increase our detection of labeled peptides? And really, we wanted to say, can a sort of more fundamental understanding of how these labeled peptides behave in the gas phase be leveraged to increase coverage of chemoproteomics detected peptides. And part of this stemmed from the fact that in nearly all of our mass spec experiments, we'd observed two ions, a mass to charge ratio of about 284 Daltons and one at about 227 Daltons, that were some of the most intense ions across the entire spectrum. And this was somewhat curious to us because they seem to be independent of the nature of the peptide, but dependent on the presence of the biotin modification. So digging into the literature, we learned that this 227 ion is likely dehydrobiotin, which is formed from fragmentation of biotin reagents, which is a fairly well-documented ion. The 284 ion was a bit more puzzling. We couldn't find any literature supporting what this might be, but we opened up ChemDraw and drew out some structures and postulated that likely this ion was this oxonium biotin species formed by fragmentation of the C5 and 6 bond, followed by cyclization to form this charged biotin species that is extremely intense across all mass spectrum. And so our next step was to really identify proteomic search algorithms that could be used to identify what we started to think of might be signature or diagnostic ions for biotinylation state. And we're really lucky as a community to have a number of labs, including Alexei Novitsky, as well as Lingjun Li's lab, who've developed algorithms that have been used to identify labile ions for glycoproteomics and really to be able to leverage the fragmentation of glycans in the gas phase, not only to identify the sites of modification, but as well to increase coverage of labeled peptides. So methods like MS-Fragger really have been harnessed to increase detection of difficult to detect modifications. And we asked the question, could we use some of these softwares to identify our ions of interest, as well as to ask whether or not they could be specific for modification state? And we also wanted to tease apart whether or not we could really support that the 284 ion was actually this oxonium biotin species. And so for this, Andrew synthesized this isotopolog 6 deuterium containing biotin azide. And when we perform a chemoproteomics with this reagent, we see instead of a 284 ion, a 290 ion. And so this, we think, at least provides fairly conclusive evidence that likely that species stems from the biotin and likely that it's either a cyclized or non-cyclized form of that fragment. The other experiment we did is we performed a MS3 level analysis where we fragmented MS2 spectra an additional time. And upon doing that, we see primary formation again of this 284 ion as well as the 227 dehydrobiotin species. And so with that information in hand, as well as using a custom FragPipe workflow, we assayed the relative intensities of the 284 ion, both without the FAMES device as well as with the FAMES device, and with a normal standard 1.6 Dalton precursor isolation window, as well as with a narrower precursor isolation window. And when we see what you can see here is with both the FAMES and the narrow precursor isolation window that we can get a good specificity of modification state. So we see that there's much more of the 284 ion and it's more intense when the peptide the precursor ion is actually identified as modified by the biotin. And so this, we think, at least begins to build the case that this ion is could be useful as a signature ion for biotinylation state. Other key pieces of information returning to this idea of trying to increase coverage of detectable cysteine-containing peptides is we assembled a panel of reagents, including those shown here, and we compared their production of fragment ions as well as detection of labeled cysteines. And what we find is, in contrast with our standard reagents up here, autosediment alkyne and biotin azide, if we flip the orientation of the azide and the alkyne, or if we increase the length of the linker for the biotin azide by 1 carbon, we can actually significantly increase the numbers of PSMs as well as peptides detected in single-shot chemoproteomics experiments. And we think this likely can be rationalized from the fact that the formation of this 7-member biotin oxonium species is less favorable compared to the 6-member species. And furthermore, for the flip of the azide and the alkyne reagents, likely the fragmentation of these species is less favorable. So in the last few minutes, I just want to return to these original questions about the cysteinome. And I think at least we've given you some things to think about in terms of trying to expand coverage of potentially druggable residues. And then to dive into the question of functionality, I have 2 short vignettes to start to access which cysteines might be functional proteome-wide. And really what I mean by functional is thinking about whether residues are involved in catalysis or disulfide bond formation, functioning as redox sensors, potentially as metal coordinating ligands like Chu Wang suggested, or modulating interactions or some other unknown function. And as part of this story, graduate student Maria Palafox in my group took a human genetics approach and decided to leverage the wide availability of human genetics pathogenicity scores, including CAD and polyphin and SIFT, as well as others. And these are scores that basically predict whether a mutation at a codon is likely going to be damaging to a protein or an organism, and combine that with our chemoproteomics data and ask the question whether or not we can pinpoint functional sites using both approaches. And what we find here is that highly reactive cysteine residues, so these are the ones that are most pK perturbed, are predicted to be more damaging by these pathogenicity scores. And so what that gives you is an additional layer of information about whether or not a residue is likely functional. The other approach that we've taken, and this is work led by graduate student Hedda Desai in my lab, is to develop a new cysteine redox platform, what we call SP3-ROX, which we have leveraged the sample cleanup properties of SP3 to enable sort of a high throughput approach to identify redox sensitive cysteines. And this community is probably quite familiar with some of the early pioneering work by Steve Gee's lab, as well as others, identifying redox sensitive cysteines using methods like OxyCat, as well as the more recent work developing the OxyMouse a limitation of all of these approaches is they're fairly steppy and they require a number of sample decontamination steps, typically by proteome precipitation or desalting. And so we thought maybe we could use the bind and elute properties of the sp3 resin for a rapid redox platform. And HEDA set this up. And together with some innovations on the data analysis side, including trying to develop a platform in our lab using FragPipe as well as Skyline to quantify cysteine residues, she was able to assay, I think, about 4,000 unique cysteines in the redox state in a quadruplicate across Jurkat cells. And this innovation here, I would say, is really important and something that hopefully the community will adopt. A lot of the early work in chemoproteomics has been done using custom in-house quantitative softwares. And so there's not been a lot of work in the proteomics community applying proteomics quantitation software specifically to chemoproteomics applications. And there's some unique challenges, particularly multi-cysteine containing peptides that make handling quantitation of chemoproteomics experiments particularly challenging. And so what HEDA did was set up not only one, but two quantitation platforms using two very standard pipelines that are widely supported by the proteomics community, showed that the ratios achieved by both platforms are roughly comparable. And from this, identified putative candidate redox sensitive cysteines. And with that platform in hand, she applied it to identify cysteine sensitive to S-nitrosylation via treatment with S-nitrosoglutathione oxidative stressor, and identified many cysteines that probably many people in this community will be familiar with, including residues in PARC7, superoxide dismutase, as well as adaptor protein crackle, which is known to associate with BCR-ABL. And more recently, she's extended this method to assay the changes in oxidation state upon T cell activation. And this is exciting because during activation, T cells produce a fair amount of oxidative stress. Oxidative stress is actually required for cellular activation. And we've been able to identify cysteines that show both increased oxidation state as well as decreased oxidation state during this cell state transformation. So with that, I'll wrap up and hopefully I've convinced you that Suzuki-Miura cross-coupling chemistry is really an exciting bioorthogonal chemistry that can complement click chemistry, that the combination of sp3 with FAMES is enabling for improving coverage of labeled cysteines, and that our new sp3 ROX method is well suited to identify redox cysteines, redox-sensitive cysteines proteome-wide. And so this work would obviously not be possible without a number of really talented people in the group, including postdoc Jian Cao, as well as HEDA, MD PhD student Alex Sun, graduate student Sunny Yan, and Andrew Palmer, as I shared throughout the talk, and collaborators Valerie Arboleda for the human genetics work, and Alexei Novitsky for the work in the FragPipe method development. And thank you all for your attention.
Thank you very much, Carrie, for this very interesting and exciting overview. Also looking into the technical details, I think this was very clear. And yeah, so we have time for a few questions. Maybe to just start, I was actually very surprised that when you showed this low coverage of the cysteines. I think it's for some, maybe not so much in the field, quite surprising. Can you explain a little bit on the molecular reasons? That is it the accessibility of the molecules? Is it the fragmentation? I mean, you showed some examples on how you improve, but can you maybe explain a little bit more what you think is the molecular basis for this?
So in terms of sort of, we're only at 10% or so of the total system affected. So I guess there's probably 20% that are in tryptic peptides that are either too long or too short to detect using trypsin. And so we and others have played a little bit with using other sequence-specific proteases, chymotrypsin, for example, to try to bolster identification of those residues. And that helps to some extent, although there's a reason that the community uses trypsin because other proteases don't necessarily give you quite as efficient or specific a digest. I think the fact that there's a wide range of protein expression, abundance, peptide ionization efficiency is obviously a challenge. chromatography, so fractionation does help to some degree, although it's not the silver bullet. I think we've done some experiments on my lab looking at subcellular fractionation, various methods, and Aranthi Wirapana has as well, and that helps to some extent. So I think, there's not a one-size-fits-all model. I'm hopeful that, more cell lines would be the silver bullet. And so in Sunny's paper, we, went from one cell line to, I think, 6 cell lines. And when you go from 1 to 2 cell lines, you see a sort of a massive increase in detection of unique cysteines, but adding additional cell lines on above that doesn't afford the same increase beyond sort of the 2 cell lines. I mean, I also suspect that potentially more unique tissues. So for example, the OxyMouse paper, I think they identified around, again, about 30,000 unique, 25,000 unique cysteines. But potentially by looking across all tissues in an organism, that might help as well.
Okay, thank you. So then there is one more question by Stephen Verhelst. As far as I understood, SP3 works with tryptic peptides instead of only proteins. For example, chloroform methanol precipitation. How small can tryptic peptides be to be still isolated by SP3? And does the modification on these peptides have any influence?
That's a great question. And I would say, we haven't done the analysis looking at whether or not there's a bias on the SP3 in terms of peptide length. I mean, in our data sets, we routinely see peptides with, 6 amino acids. In terms of whether the hydrophobic modification helps, in terms of enriching it on the resin, I don't think it makes a substantial difference. We've done these experiments now with standard isotope reagents. So, tripeptide labeled drug, we've done it with biotin azide, we've done it with the biotin bronic acids, and they all seem to work fairly comparably. We also, the SP3, I think, little known feature of it is you can do decontamination both at the protein level and at the peptide level. So it works for binding proteins. They bind much more strongly, and so you need to use more harsh elution conditions to recover your material compared to peptides. And so I think potentially paying attention to the specific conditions for the elution to get the method to work. And I think, we've tried in my lab to be quite precise in the methods of our papers about how to get this to work, because there's some nuance. And if you set it up, it's not necessarily going to work the first time perfectly. And so, I'd obviously encourage the community, if you have troubleshooting questions, please don't hesitate to reach out.
Okay, thank you very much. So maybe very quick question, and then we have to move on by Stefan Hacker. Have you tried other cysteine probes, hyperviolent iodine, heteroaromatic sulfones, would largely increase coverage, or if you would find same cysteines as with iodoacetamide alkyne?
We have not stepped beyond iodoacetamide alkyne besides, comparing aryl versus alkyl iodoacetamide alkynes. What we have done, and I think potentially the reason new probes would actually help is not because they would access cysteines that weren't labeled by iodoacetamide alkyne, but because they might change the physicochemical properties of the labeled peptide such that it would co-elute in a region of the MSMS spectra that was a little bit less rich and therefore biased towards detection. Because we have done experiments where we've essentially capped peptides with iodoacetamide alkyne and then just digested the samples and not enriched and looked for the percentage labeling. And iodoacetamide alkyne and iodoacetamide in general caps, upwards of 98% of all cysteines. So I think, again, it also depends on if you're labeling in a native or just denatured state. And we've switched mostly labeling in denatured state to really cap all cysteines with equal efficiency.
Okay. Great. Thank you very much, Carrie. So I think we have to move on. Thanks a lot. And our next speaker and final speaker of the second keynote today is Carolyn Bertozzi. Karen, I think, you already shared the screen. Thank you very much. So Carolyn Bertozzi from Stanford University, she doesn't need much introduction. Her pioneering work on bioorthogonal ligation is now textbook knowledge. We teach it actually in our university classes, so everyone is familiar with your work on this. I think what's really exciting is that today you talk about a new topic, which is targeted degradation of extracellular proteins with lysosome targeted chimeras. And we are looking forward to this presentation. And without further ado, I would like to hand over to you to listen to your talk. Thank you very much for coming, and stage is yours.
Thank you so much, Stefan. It's really great to be here, and it's an amazing lineup of speakers, and I'm just very happy to be among them. And the title kind of speaks for itself. I thought I would tell you about a fairly new project in the lab that basically is aiming to develop therapeutic molecules that do for proteins on the outside of the cell what we are familiar with PROTAC-like molecules doing on the inside of the cell. So first, though, it's incumbent upon me to declare my conflict of interest. So I have a startup company called Lycia Therapeutics that has an exclusive license to a patent that covers this technology. Okay, so I'm going to tell you about the work primarily of two people, a former coworker, Stephen Bannock, who was a postdoc that launched this project now 3.5 years ago, but since then has moved to his own assistant professorship in my own department at Stanford University, where his lab is taking off like a rocket ship and where he continues to develop innovative new therapeutic modalities based on induced proximity, as well as a late-stage graduate student named Green An, who's in her now 5th year in the PhD program and will be wrapping her work up over the next year. So, really the story does start with the PROTACs. This is a concept that came out of the academic labs of Craig Cruz and Ray Deshays with a landmark paper published back in 2001. And here we are 20 years later, where every major biopharma company and many small independent companies are focusing entirely on this exciting new modality for medicinal chemistry. And, the notion here is that there are certain targets which are difficult to drug using ligands on their own. But if one can convert a ligand to a degrader, there's an entirely different type of pharmacology that can be induced. And the way that was done for the PROTACs, or the proteolysis targeting chimeras, was to fuse the ligand that interacts with the target protein with another ligand that recruits an E3 ligase and basically targets that protein to the ubiquitin proteasome system for degradation. And earlier we heard Chris Parker develop a really interesting and creative new take on this strategy, where instead of targeting a protein for degradation, he's targeting proteins for post-translational modifications. And so, that's a harbinger of many creative new applications of this induced proximity concept yet to come. But for degradation, we were sort of watching this field unfold from a distance and kind of envious of the fact that people were developing degraders of intracellular proteins, but that kind of left a whole bunch of proteins on the shelf. And in particular, those are the extracellular proteins. And for me, as someone who's spent her career largely studying glycosylated molecules on the cell surface or in the extracellular space, many of the targets that we were interested in were inaccessible to PROTACs because they're not co-located with the ubiquitin proteasome machinery, which is here in the cytosol of the cell. And in fact, the estimate is that about 40% of the proteins that are encoded in the human genome lie in this extracellular space, predominantly or entirely. And this includes many membrane-associated proteins that don't have a cytosolic domain that is amenable to small molecule ligation, as well as proteins and particles and aggregates of proteins that are entirely secreted and in the extracellular space that you would like to get rid of in a therapeutic modality. So this includes, for example, receptor tyrosine kinases on the membrane that have been difficult to drug using more conventional approaches, various adhesion molecules, immunomodulatory receptors that when you ligate them, often you end up activating them rather than blocking them. And then soluble targets like cytokines, immune complexes, amyloid aggregates in the periphery that can deposit in tissues and cause pathologies. These are all the kinds of targets that, you could make an argument are good for a degradation strategy, if only there was one for the extracellular space. So when Steve joined the lab, he started thinking about what are the machineries for degradation that these extracellular molecules are most often processed through. And it happens to be the endosome-lysosome system. So the lysosome is an organelle that is packed full of enzymes that will degrade virtually any type of biomolecule. So lysosomes have proteases, glycosidases, RNases, lipases, and so on and so forth. So if you can get a biomolecule into the lysosome, there's a good chance it will be degraded there. So the way this happens in nature is there's a system to internalize cargo into endosomes, first an early endosome, which then undergoes a maturation process to eventually become a late endosome, which fuses with the lysosome through this endolysosome fusion process, and the cargo ultimately is degraded therein. And there's a classic pH change that occurs throughout the course of this maturation process, so that outside the cell, the pH is around 7. And then once the endosome matures all the way to the lysosome, there's a gradual drop in the pH, ultimately to even less than 5. So how is it that cargo gets selected to be basically trafficked through this pathway into the lysosome? Well, there are about a dozen known lysosomal trafficking receptors that can engage with different cargo and shuttle them through the system. And when you study glycoscience, there's one of those receptors that you learn about in chapter 1 of your book, and it's the mannose-6 phosphate receptor, which serves the job of shuttling lysosomal enzymes into the lysosome, but can also capture cargo from outside the cell. So M6PR has this really interesting recycling and trafficking behavior. It shuttles between early endosomes and the Golgi compartment, where lysosomal enzymes are being biosynthesized, they're glycosylated, they're modified with this mannose-6 phosphate mark, which is the red dot, and captured and then trafficked to the lysosome where they can perform their functions. But at any given time, about 10% of the mannose-6 phosphate receptor resides on the plasma membrane. And if molecules from outside the cell carry the mannose-6 phosphate mark, they can be captured and pulled in through endocytosis and basically dropped into the lysosome. And there's a pH switch on the binding of this receptor to mannose-6 phosphate. And as the pH drops, the affinity goes down. And that's how this receptor can capture cargo, deliver them, release them in the endosome lysosome system, and then recycle back either to the membrane or to the Golgi compartment. So we had the idea that we could hijack this receptor to deliver cargo into the lysosome for degradation. And the way we would do that would be through the intermediacy of a heterobifunctional molecule that we call a lysosome targeting chimera, or a LITAC. So the two parts of this molecule would be one part that binds a target protein of interest that resides in the extracellular space, and the other part would be a binder for the mannose-6 phosphate receptor. So this molecule would be a bridge between the protein of interest and this lysosomal trafficking shuttle. And in this cartoon, I'm illustrating the target binder as a monoclonal antibody, and that most of the LITACs we have made so far are based on known antibodies. It's a convenience, but one should think agnostically about this binder. It could be an antibody fragment. It could be an aptamer, a peptide, a small molecule. And since our initial publications in this space, we've made LITACs based on all of those different kinds of molecules. And then in our initial molecules, the mannose-6 phosphate receptor binders that we used were glycopolypeptides, in which we had modified the peptide backbone with mannose-6 phosphate analogs. And so the way this worked was a target of interest, whether it was soluble or membrane associated, would bind to this binder, to the antibody, and then on the same cells, this mannose-6 phosphate receptor binding component would interact with the receptor and basically allow trafficking of that target into the lysosome, followed by degradation. And in our initial conception, we were pretty confident that soluble targets would be amenable to capture and degradation through this pathway, for the simple reason that there was ample precedent in the literature and in biopharma industry for delivering lysosomal enzymes to the lysosomes through external administration. And in fact, this is how enzyme replacement therapies work. So there are rare diseases where people are lacking lysosomal enzymes, and you can infuse those patients with recombinant forms of those enzymes that have the mannose-6 phosphate mark on them, and they will just naturally get taken up by cells by this receptor and trafficked to the lysosome, and that works. What we were a little less certain about is whether a membrane-associated molecule could be hijacked in cis and delivered to the lysosome through the mannose-6 phosphate receptor on the same cell. That is, to our knowledge, not a natural biology, and so that was a little bit more of an open question. But you have to do the experiment. So what Steve did in his initial pass is to synthesize these glycopolypeptides with mannose-6-phosphate groups using a nickel NTA polymerization chemistry that had been developed many years earlier by Tim Deming's group at UCLA. And this was a chemistry we were using for other purposes, having nothing to do with Litax. So it was kind of just a bit of a convenience that we had the knowledge for how to make these glycopolypeptides very quickly. And one of the benefits of these polypeptides is it's a quick way to have multivalent display of an epitope like mannose-6-phosphate, which from some literature we thought would boost the avidity of these reagents for the mannose-6-phosphate receptor and maybe kind of drive their internalization more efficiently than monovalent ligands might. So this is how the chemistry worked. Steve took mannose through 11 steps that I won't go into. He converted it into a protected form of a mannose-6-phosphonate, which has a carbon atom rather than an oxygen atom here. And that renders these molecules resistant to phosphatases. And there's the NCA group. It's this N-carboxy anhydride. So this is a monomer for the polymerization chemistry. And it's a living polymerization. So the length of these polypeptides can be controlled by the ratio of the monomer to the catalyst, which is a nickel catalyst. And typically in these early molecules, Steve would mix together mannose-6-phosphonates with a spacer amino acid like alanine, just to kind of decongest the sugars along the polypeptide backbone. So these polypeptides had a free amino terminus, and we used that site to conjugate them onto antibodies. And so using chemistries that, again, we're very familiar with in our lab, what he did in this first pass was to take antibodies of interest and just non-specifically modify their lysine residues at sort of a substoichiometric ratio with an NHS ester molecule, with little peg spacer with an azide group, so that he could then click the glycopolypeptides onto those azides by simply modifying the amino terminus with commercially available bifunctional reagents that have a cyclooctane on them. And this is all published chemistry, so I'm not showing the details. But in this first generation of Litax, a typical preparation would produce Litax that had 10 mer glycopolypeptides, and on average, 3 of these glycopolypeptides attached to lysines heterogeneously. Okay, so we considered these the sort of quick and dirty first generation Litax. And it's kind of fun to follow the chemistry by native gel analysis, because you can see how the starting antibody, usually runs as a mixture of bands because of the glycoform heterogeneity. But then after doing the lysine modification chemistry with an azide spacer, you get an increase in the net negative charge, having acylated positively charged lysines. And then after conjugation of these mannose-6-phosphonate glycopolypeptides, the net negative charge is even further enhanced, which increases the migration in the native gel. And again, these early stage Litax were pretty heterogeneous, and they had a lot of mannose-6-phosphonate groups on them, typically 30-ish per antibody. As I'll show you later, that turned out to be overkill, but these were the first molecules that we made. So to test whether they could mediate the clearance of a cargo through the mannose-6-phosphate receptor, we did a simple in vivo experiment where we pre-complexed either a Litac antibody or the naked antibody counterpart with a fluorescently labeled cargo protein, which in this case was another antibody. And this was sort of a model of an immune complex, which is an interesting therapeutic application of the Litac technology. And then what Steve did is he injected either the Litac or just the naked antibody complex into mice. And then after periods of time, he would draw blood and look at tissues and just quantitate by fluorescence gel the amount of that cargo antibody that remained in the system. So it was a rabid IgG with Alexaflor 680 on it. And we could quantitate the amount of that cargo protein just by running a gel and analyzing the gel on the Li core. And so what you can see is the column here with the Litac minus, those are the complexes with naked antibody, where Litac plus are the complexes with the Litac antibody. And after 24 hours, already there's some clearance of that Litac complex versus the naked antibody complex. And after 48 hours, it's almost entirely cleared from the system compared to the native antibody complex. And that was true both in serum as well as in 2 different tissues that we looked at, the liver and the spleen. So again, this was sort of as we had hoped and expected based on known literature of lysosomal enzyme uptake out of the system. But the membrane proteins were a bit of a more of a wild card. So to look at a membrane protein, we chose the epidermal growth factor receptor as a model target of interest. It is an important cancer target against which several drugs have been made and approved and are important standards of care for various different indications, most prominently lung cancer and colon cancer. And there's an antibody that's an approved drug against EGFR called cetuximab. So cetuximab binds EGFR and blocks the binding to its ligand, which is epidermal growth factor, but it doesn't affect the total levels. So what we did is we made a LITAC out of EGFR, just using the chemistry I previously showed, to see whether we could convert cetuximab into an antibody reagent that now degrades EGFR. And that worked stunningly in our very first experiment using HeLa cells, which express EGFR at very high levels. So what you're looking at are the fluorescence micrographs of these HeLa cells, in which we can label EGFR with an antibody that binds an independent epitope, and it's showing us the pink fluorescence. So untreated HeLa cells have EGFR populating the plasma membrane at high levels. When you treat those cells with cetuximab, there's no change in the distribution of EGFR, still all over the cell surface. But now when there's the mannose-6-phosphonate glycopeptides on cetuximab, we drive EGFR into the lysosomes, and you can see a dramatic redistribution of that protein. And importantly, it is getting degraded. So we can quantitate EGFR levels by Western blot, normalizing to a standard housekeeping protein. And in red, you're seeing changes in the level of EGFR as a function of time after one dose of the LITAC reagent versus the change, which is not much, in the abundance of EGFR upon treatment with the naked antibody. And so in this experiment, we basically were resetting the steady state levels of EGFR on these cells down to about 20% of what they normally would be. And we really can't go to zero in an experiment like this, because while we're driving the degradation of EGFR, the cells are always making new EGFR. So basically the flux of EGFR has changed by virtue of having the LITAC present. An interesting question that came up in our mind is, membrane proteins have their own endogenous recycling and turnover biology, right? They're being made and degraded in certain ways at certain levels. And for some proteins, they're engaging in a recycling biology that sustains them on the plasma membrane in a very active way. And can a LITAC compete with an active plasma membrane recycling biology? So a great example of that is the very famous immune therapy target PD-L1, which is the target of Roche and Genentech's drug Tecentriq, right? So this is the ligand for the checkpoint receptor PD-1. And PD-L1 is maintained at high levels on cells by virtue of an interaction with a chaperone protein called CMTM6, which is this green squiggle. So when CMTM6 is expressed, it interacts with the cytosolic tail of PD-L1 and basically drives it through the recycling endosome system so that it stays on the plasma membrane in preference over getting basically degraded through the endosome-lysosome pathway. So the question is, under that circumstance, could a LITAC that binds to PD-L1 redirect it for endosome-lysosome trafficking by virtue of now the engagement with the mannose-6-phosphate receptor? And who might win that battle? And so we made a LITAC out of Tecentriq, and what we found is that indeed the LITAC is able to reset the steady state levels of PD-L1 by driving degradation in competition with recycling biology. So what you're looking at here through Western blot are the levels of PD-L1 on cells on the left-hand side that were treated with either the antibody alone or different LITAC reagents. And on the right, it's the same kind of experiment, but now we have added a competitor for the mannose-6-phosphate receptor, which is 5 millimolar of mannose-6-phosphate. And all you really need to see is basically this fourth lane on the Western blot. That's the Tecentriq mannose-6-phosphonate LITAC. Everything else is a control. And we've reset the protein levels from 100 at the start down to about less than 30% of steady state in the presence of the LITAC. And there's no reduction that's significant when the LITAC is outcompeted with mannose-6-phosphate. So when Green came into the project as a grad student, she was excited about the early work with these first generation LITACs, but she was frustrated with basically how heterogeneous their structures were. As this audience knows full well, when chemistry produces a heterogeneous product, it's really hard to optimize the structure because you can't really do structure activity relationship studies. And so Green's first mission as a graduate student was to basically design new chemistries to make Litacs that were more homogeneous and therefore more optimizable. And she started with a focus on the structure of the mannose-6-phosphate receptor ligands. And so whereas Steve had used this polymerization chemistry to very quickly bang out polymers that had multivalent mannose-6-phosphate display, Green decided instead to make glycopolypeptides homogeneously through solid phase peptide synthesis, and then she would have to make them much shorter for this to be pragmatic. So just to compare the difference between the two ligands, the NCA polymerization gave us glycopolypeptides that were anywhere between 10 and 90 monomer units, which we could bias based on the initiator to monomer ratio in the living polymerization, but the polydispersities were typically 1.1 to 1.2. So there was moderate control over the length and composition, but they were heterogeneous. Also, because of their heterogeneity, they were difficult to characterize and also really kind of difficult to scale up, which we thought would be important to do more in vivo studies. By contrast, simply making these by solid phase peptide synthesis allowed Green to control precisely their length and composition. They were homogeneous, easy to characterize, and amenable to scale up. And in her first pass, what Green did is she just made two different polypeptides, one that had 2 mannose-6-phosphonates and one that was a little longer with 5 mannose-6-phosphonates linked through serine residues. Okay, so these are very simple, like dipeptides and pentapeptides. And she conjugated them to cetuximab using the same chemistries we had used earlier. So there was still some heterogeneity because they were modified on lysines, but at least the ligands for mannose-6-phosphate receptor were now much cleaner. And the good news is that even with these much lower valencies of mannose-6-phosphonate per antibody, she was able to make very effective Litacs. So here's an experiment where she took the pentaglycopolypeptide cetuximab conjugate and showed that it could degrade EGFR in a cancer xenograft model using the HCT116 cell line, which is a colon cancer-derived cell line. So in this experiment, she grew the tumors subcutaneously in immune-compromised mice. And then when they had grown to a certain size, she went through a dosing regimen where she injected these animals with 2 mg per kg of different cetuximab reagents on 2 different days, so 2 different doses. And then on the 3rd day, she harvested the tumors, ground them up and lysed them, and then quantitated the level of EGFR by Western blot. And for 2 different experiments with 2 different groups of mice, what she found is that she could get degradation of EGFR, a reduction in the steady state level with just this, pentaglycopolypeptide cetuximab conjugate versus naked cetuximab on its own, which doesn't have any effect on the level of EGFR. So we'd gotten one step closer to having cleaner molecules that still are effective Litacs, despite the fact that there's a lot less mannose-6-phosphonate groups per antibody than in the first pass molecules. Also, Green went on to show that she could kind of replicate this type of activity on Litacs made from lots of other kinds of antibodies that bind different targets. And I'll just show one example. c-Met is a really exciting oncology target. It's a cell surface receptor tyrosine kinase that has proven to be quite difficult to drug using small molecule kinase inhibitors or antibody-like reagents on their own. And as an example, Genentech made a single-arm antibody called onartuzumab, which actually has been in human clinical trials, but didn't really have the efficacy that they were looking for. It's a single-arm antibody because more conventional double-armed antibody has the unfortunate consequence of activating c-Met rather than blocking it. So we took onartuzumab and converted it to a Litac using those, pentaglycopolypeptide ligands that I had just shown. And, at least in cell culture, it looks like this Litac is able to reduce the steady state levels of c-Met down to something like, maybe 20% of wild type. And you can see that here's the Western blot, here's the c-Met levels. The cells are treated either with the naked antibody or with onartuzumab Litac. And here's quantitation of the reduction in cell surface c-Met, both on HEP3B cells and UMRC2 cells. So we're really interested to take these onartuzumab Litacs into tumor models and see if we can degrade this hot target in vivo. Meanwhile, the mannose-6-phosphate receptor was a great starting point for Litacs might not have mentioned that it's expressed on virtually every cell type. It's kind of a housekeeping protein in mammals. So you can pretty much count on mannose-6-phosphate receptor to be there wherever you are and wherever you want to degrade a target. But there are other lysosome targeting receptors that are more tissue specific. And one that we thought would be fun to look at is the asialoglycoprotein receptor, or the ASGPR. Which is a very efficient lysosomal trafficking shuttle that is very specifically expressed in liver, in hepatocytes. It has very high copy numbers there, so 1 million per cell or more. And it has the fundamental job of clearing serum glycoproteins out of circulation through basically clathrin-coated, PIT-mediated endocytosis. It binds to either GalNAc or galactose, and it's called the asialoglycoprotein receptor because when sialic acids are removed from the typical glycan on a circulating protein, it reveals an underlying GalNAc or galactose, and that's a sign that that glycoprotein has aged and needs to be cleared out of circulation. And this is a very familiar liver trafficking receptor for people in the oligonucleotide therapeutic space. This is exactly the receptor that Alnylam and Ionis hijack in order to get very efficient liver targeting of their medicines. And I'm just showing the structure of one of those asialoglycoprotein targeting moieties that's used at Alnylam. So there's a little dendrimer with 3 GalNAcs on it that the Alnylam group attaches to their siRNAs. And this is now a component of approved drugs and really allows very efficient accumulation of these oligonucleotides in lysosomes and hepatocytes. So we just borrowed this exact moiety and used it to see if we could make Litacs that could capture either soluble or hepatocyte membrane glycoproteins and target them for degradation through this system. So Green synthesized that Alnylam dendrimer, functionalized it with a cyclooctine reagent, and then clicked it onto antibodies that had been pre-functionalized on their lysine residues with a little azide tetrapeg spacer. And in her first generation GalNAc Litacs, she made some really high valency Litacs where she would have on average about 11 of these dendrimers per antibody on the lysine residues. And each dendrimer has 3 GalNAcs. So these are like 30 something GalNAcs per antibody. And as you'll see, that also turned out to be overkill, but these were the first generation molecules. So we tested them as Litacs for EGFR, and again, they were made from cetuximab, using a variety of hepatocyte cancer cell lines that express both M6PR for the mannose-6-phosphonate Litacs, as well as ASGPR for the GalNAc Litacs. So these cell lines are great because we could do kind of a head-to-head comparison for the 2 different lysosomal trafficking enzymes and see how they compare, against the same target, which was EGFR. And as you can see in these systems, you're looking at the basically normalized total EGFR levels on cell lysates. And the results are pretty much the same for every cell line, that both the GalNAc Litac from cetuximab and the mannose-6-phosphonate Litac are sort of equally able to reduce the steady state levels of these proteins down to about either between 20 and 40% of the wild type levels. So they're quite comparable on cell lines in cell culture in terms of their ability to drive degradation of EGFR. But when we went in vivo and looked at their ability to reduce the level of certain targets in tumor models, the 2 different Litacs performed quite differently. So this was a xenograft model with the HepG2 cell line, which was one of the cell lines on the previous slide. And in this case, instead of EGFR, we were looking at HER2, which is of course a very important oncology target. And Green had made Litacs from a clinically approved drug called pertuzumab, which binds HER2. And she made those Litacs either with mannose-6-phosphonate glycopolypeptides or with these GalNAc dendrimers. And then she did a model where she grew the tumors in mice, and then she dosed them with the various pertuzumab conjugates, 1 dose per day, 3 different days, harvested the tumor, ground them up, and then looked at the levels of HER2 in a Western blot. And so what you're looking at here is the HER2 from these xenografts with saline-treated control mice, and then compare those to pertuzumab mannose-6-phosphonate Litac, or pertuzumab GalNAc Litac. And you can see that pertuzumab on its own as a naked antibody actually reduces the level of HER2. So it looks like this antibody is able actually to drive some reduction maybe internalization and degradation. But you get a whole lot more degradation and reduction of HER2 if you treat these animals with a pertuzumab mannose-6-phosphonate LITAC. But interestingly, the pertuzumab GalNAc LITAC was totally ineffective at driving degradation of HER2 in this model, even though it was equally effective as the mannose-6-phosphonate LITAC on the cultured HepG2 cells. So what went wrong here? And we suspected that the GalNAc LITAC was not as effective in vivo because we had put so much GalNAc on it that maybe it was just getting very rapidly cleared from circulation through ASGPR in the liver. And so to address that, we thought we really have to reduce the valency of GalNAc molecules on these antibodies. And a way to do that is to switch over from this sort of non-specific lysine chemistry, which is difficult to control, to a very specific, site-specific, in fact, method for modifying these antibodies. And so years earlier, we had developed a strategy for site-specific protein modification that we had applied to antibody drug conjugates. And it used a method that we call the aldehyde tag method. And I'm not going to go through the details because that's all published work. But it suffices to say that we can genetically